Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Abdelrahman Al-Anani, and inshallah I will be presenting to this session. I want to say that I made sure inshallah to include everything in this crash course, so inshallah it will be enough for physiology. Also I will put the slides in the description, so if there's like anything that you want to check by yourself or read or do whatever, you can take the slides. And I will also put timestamps, so if you want to visit a certain lecture, please feel free to do that. Also, this is my phone number and my email. Don't hesitate to reach out to me if there is any question or anything that you didn't understand. Before we start, just a quick roadmap. We will be going through the lectures like the same order they are in the schedule. The only difference is that I moved P8 to the end, you see? Because why is large intestines P8? I do not understand that, so I took the matter in my own hand. And let's start with the first lecture. Basically, these are the general functions of the GIT. No one will ask you, tell me what are the functions of the GI tract, so you don't need to memorize them. Just know or keep in mind the food processing, because we will be talking about it now. Food processing. It could be many different things. It could be either digestion, and digestion is the breaking down of large molecules into small molecules, so they are easier to absorb. For example, you like proteins. You turn them into amino acids by digestion. Why? So you can absorb amino acids. It's easier to absorb. Now, what's absorption, aslan? When you take these small molecules that you have just made by digestion and take them into the bloodstream. This is called absorption. Okay? From the lumen of the organ, of the GIT organ, into the bloodstream. Now, secretions. Secretions could be many different things. Either you secrete something from the cells of the wall into the lumen, like hydrochloric acid, for example. You are secreting hydrochloric acid from the stomach into the cavity of the stomach, right? Or it could be paracrine secretions, which mean it affects neighboring cells. We will be talking more about that later. Don't worry if you don't get it now. And also endocrine secretion, which means that it's secreted from the cells into the bloodstream directly. Like hormones, you know hormones. So we'll be talking also about that. Motility is the movement of food from up to down in the GI tract. So you see when you eat something, it goes from up to down, right? This movement of food is called motility. And we will discuss it also later. Now, the histological structure of the GI tract. I know this doesn't sound like physiology, but we will need it for what's next, okay? So please bear with me. When we are talking about a tubular organ, any tubular organ, it has this general structure of mucosa, submucosa, muscularis, and adventitia or serosa. No one cares about this. So what's important about this? I want you just to keep two things in mind, okay? The muscularis layer here is actually not one layer, it's 2 layers of muscle, okay? 2 layers of muscle. And in the stomach it's 3, but let the histology be able to tell you about that. This is not our concern now. Just I want you to keep in mind, we have what? 2 layers of muscle in the muscularis layer. And we also have another layer of muscle, which is this thin layer that's found in the mucosa, called muscularis mucosa, from the name. It's in the mucosa and it's muscle, so it's muscularis mucosa. Okay? Just keep this in mind and we will talk more later. Okay, so when we talk about regulation of GIT functions, it could be either nervous or hormonal. Regulation of anything is nervous or hormonal. If it's nervous, I want you to keep in mind that it starts strong from the mouth. So when we talk about saliva or anything like that, it will be mostly nervous control, okay? And it decreases from mouth to anus. So it gets less important while you are what? Moving from up to down. Hormonal is the exact opposite. It starts very weak in the mouth and it becomes stronger the more you go towards the anus. It's just as a general rule of thumb that you should keep in mind. Okay, now we will talk about nervous control specifically in this lecture, and it can be divided into 2, either extrinsic control or intrinsic control. What is this and what is this? We will talk about it now. Before we talk about it, I just want to note that whenever you see this starfish in any slide, that means that there has been a question asked about this slide before, either in the 17th batch final exam or in Dr. Angelica's practice questions, okay? So, yeah, I wouldn't, of course, advise you to only study that, but just keep it in mind. Now, we said that there is extrinsic and intrinsic control of nervous system here. What do we mean by that? Extrinsic control is the same control that's found in any other organ other than the GI tract, which is sympathetic, parasympathetic. We already know that from many other systems that we have studied. So we have this sympathetic and parasympathetic. Okay? Now, the intrinsic control is different, and it's very special to the GI tract itself. It's called the enteric nervous system, which is basically nervous plexuses, which is collection of neurons when we say plexus, that are found within the GI tract organ wall. So if you take the stomach and you look at its wall, you will find these neurons or collections of neurons. We call it enteric nervous system, and we will discuss it even more later. But I want you now to keep in mind something. This sympathetic pathway, which is coming from the T5 to L2 vertebras, is the same as anything that we have taken before. Pre-ganglionic, post-ganglionic, and it's easy to understand, right? You have this pre-ganglionic going, synapses in the ganglion, and then gives us post-ganglionic, and it goes to the organ. Easy. Now, Now, what's different is the parasympathetic. Why? You see, it's only pre-ganglionic. This vagus is pre-ganglionic, and these sacral nerves are also pre-ganglionic. So where is our post-ganglionic parasympathetic? The post-ganglionic parasympathetic are actually the enteric nervous system neurons themselves, and we will discuss it in 2 minutes or something, okay? But just keep this idea in mind, because the doctor didn't, unfortunately, she didn't explain this idea, and it's very important, okay? She asked a lot of questions about it, but she didn't explain it. Now you will see that we have this cross-section of the esophagus, okay? We have the esophagus, we cut it in half. So this is up and this is down, okay? I want you to keep this in mind. We rotate it this way, so now it's up and down. We zoom in the wall, and this is what we find. So what's going on? You see the star, huh? So that means that it's very important. So what's going on here? This is the lumen of the GI tract. Here it's the esophagus. We took it as an example, right? And please don't forget that this is up and this is down. So the food is moving this way in the lumen, and it's stretching the wall here. Who's feeling this stretch? These sensory neurons. Can you see them? So the stretch signal goes to this sensory neuron, and it moves in two pathways. One moving up and one moving down. Remember, we said that this is up and this is down. Okay. So this signal is going up and down at the same time. Why? The signal that's moving up is going to these excitatory motor neurons. The signal that's going down is going to these inhibitory motor neurons. What's happening? Excitatory neurons that are found up are gonna cause contraction of the muscle. So contraction, where? Up the upstream of the food that's stretching the wall. Now the inhibitory neurons that are found downstream will relax the muscle. So what did this lead to? Before the food there is contraction. After the food there is relaxation. So the food is being pushed from up to down, up to down. And this causes the movement of food from up to down in our body. And this we call peristalsis. Okay? I hope that makes sense, because it's a very important concept to grasp one. This is why even if someone is eating upside down, please don't do that. But if someone does it, the food will still go to the stomach, because it's not gravity only that's doing the work. It's actually the organs themselves, they are squeezing themselves up to down. Now, these collections of neuron all over, like the sensory, the inhibitory, the blah, 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 all of these guys, okay, are called Auerbach's plexus. The guy who discovered them was called Auerbach. And they are also called myenteric plexus. Why? Because they are found between the two layers we talked about in the histological structure. Do you remember when I said the muscularis here is 2 layers? In between we have these plexuses. Okay? Makes sense? Makes sense. Another plexus here that's found also, and it's, the food is stretching the wall. So the sensory neurons takes this signal, and it goes directly to this secretomotor neuron. It tells the cells that are responsible for secretions to start secretions, like glands, for example. to increase their secretions. Okay? And we call this neuron plexus Messner's or submucosal plexus. Submucosal because they are in the submucosa, and Messner is the guy who discovered them, just like Auerbach. Okay? I hope that makes sense. Now, something that you can interpret here is that this whole thing, like from the stretch to the neuron, to the contraction, to the secretion, all of that is happening within the GIT organ wall itself. So here is the esophagus. It's happening in the wall of the esophagus. We didn't leave from here. Everything is happening inside. So these neurons are, so these plexuses are what we call the enteric nervous system that we talked about before. And they are considered independent of the central nervous system. So even if you cut the biggest nerve or whatever, they will still be able to function. But at the same time, can you see these guys? They are also neurons and nerves of the central, of the autonomic nervous system, which are affecting the rate of work. So for example, they can increase the secretion, decrease the secretion, so they can control them, right? The central nervous system can control it, but it's still independent, which means it doesn't need it to function. I hope that makes sense. Now, the next slide is just a summary, and you can see there is 2 stars here because this is a very important slide, and I found like 13 questions about it or something. So the enteric nervous system, it's present the GIT from esophagus to the rectum. We just took the esophagus as an example. Independent of the central nervous system, but can still be controlled by it. That doesn't mean it doesn't need it at all. It contains the 2 plexuses. We talked about all of them. You can please stop and read this if you want, because it will help you grasp the concept very well. Just a quick thing about the neurotransmitters. What makes these cells excite and these cells inhibit? Why? What's different about them? It's the neurotransmitter that they release, okay? So acetylcholine is responsible for contraction. So the excitatory cells that we talked about release what? Acetylcholine. And VIB and nitric oxide are responsible for relaxation. These peptides are what, or neurotransmitters, are what cause this difference. Now we have short and long reflexes. This is what we have talked about. Short reflex is when it's independent, like these guys. So it goes, like the signal goes from the sensory neuron to the excitatory neuron, and everything is happening within the wall of the gut itself, right? This is called short reflex. If there is control from the central nervous system and the signal, instead of going this way and stopping, it went to the central nervous system, and we had an order from the, for example, the brainstem or whatever to increase the work, decrease the work, or whatever. This we call long reflex, so it involves central nervous system. Now, Hirschsprung's disease, it's basically when part of the GIT doesn't have these neurons, which are called Auerbach's plexus. These plexus, sorry. These plexuses, if they are not found, it's called Hirschsprung's disease. What's the problem? You see, here there is constriction of the rectum. Why? Who caused the relaxation? It was the neurons of the Auerbach's plexus, right? The inhibitory ones that release VIP or whatever to relax it. Here we don't have them. So what's the problem? Constriction of the rectum. The feces cannot come out. And you see the section above the rectum right away, which is here, sigmoid column, for example, it's very, very, very dilated. Can you see that? Why is that the case? Because feces are accumulating here. They are trying to go out, but the rectum is stopping them, so you have this dilation. What's the solution? Cut it out. This is the only solution. And it's called Hirschsprung's disease. Okay? For clinical cases, I will not be discussing them unless there's something that I didn't mention that I need to add. But you will have the answers. You can read them yourself if you are interested. Or, of course, if the clinical case is also can be asked about in the SAQs, inshallah, I will be also talking about it. But this one is not very important. A few questions, and then we are done with this lecture. So, which combination of afferent and efferent neurons locations characterizes short loop reflex in the GIT? Short loop means that we are where? Okay? Short loop means we are inside the wall, so only the plexuses themselves. And it's going to be A, parasympathetic ganglion, the intestinal wall, and also parasympathetic ganglia in the intestinal wall. Why? Because we said that enteric nervous system is considered to be the postganglionic parasympathetic, if you remember. So these neurons are found where? In the parasympathetic ganglia itself. Okay? And whether it was afferent or efferent, the sensory or the motor neuron, they are all within the GIT wall. Why? We said it's what? Short loop reflex. Now, which of the following best describes the role of the enteric plexus? Okay, both of you wanna solve it yourself? Let's see. Coordinates, peristalsis, and motility. We discussed that. Okay, so second lecture. Let's start. So we have these GI peptides. What do we mean by GI peptides? They are basically things that are secreted from the GIT organs or cells, and they cause different, or they have certain functions, okay? We will talk about them in details, but they are divided generally into three main groups: classic hormones, neurocrines, and paracrines. Classic hormones means they act like hormones, they go through the bloodstream, endocrine secretion, basically. The other ones we will talk about when we come to them, okay? So we have these classic hormones, as we said, they are released, this is also, you should know, they are released by APUD cells. They are basically cells responsible for these hormones. And what are the examples? Gastrin, CCK, secretin, GIP, motilin. We will talk about each of them now in details. By the way, this lecture is heavy in memorization. I will try my best to help you with it, but please know that you will have to memorize quite a lot, okay? Now, before we go to gastrin, I want to set up a few rules with you guys. When you think about a hormone, ask yourself about 4 things about this hormone. Where is it released from? Where is it released from? Second thing, what stimulates this hormone secretion? What inhibits this hormone secretion? And last thing is, what effects does this hormone do? Again, where, what stimulates, what inhibits, and what effects does it do? Okay? Okay. So for gastrin, first of all, where? It's released from G cells, from the antrum and duodenum. Antrum is a part of the stomach. Okay? Now, there is an additional note about gastrin, is that it shares the same receptor with CCK, and we will discuss that later when we go to CCK. Now, second thing, what stimulates the secretion of gastrin? To understand gastrin, you should know what its goal is. Why is it doing what it's doing, okay? And the short answer is gastrin wants to digest food that's already in the stomach, and it wants to empty the stomach from that food. Again, it wants to digest the food and empty the stomach from it. So now it will make a bit of sense when you see why is gastrin secreted. It will be secreted when there is a smell, sight, taste, thinking of food, anything that relates with food, it will release gastrin. Why? Because it wants to digest food, right? So when you are anticipating food, vagus will cause the release of gastrin. How? We will talk later. Distension of releasing sites. So if the antrum or the duodenum are distended, gastrin will be secreted because you want to digest the food that's there. Protein digestion products also make sense, because come on, like you have protein that's partially digested, it means that you want to digest it more, so you release gastrin. Inhibited by what? High acidity in the releasing sites, and it will make sense when we say its effects now, its effects. And hyperosmolarity of common fats in the duodenum, you will also see. Here, this is what we talk about, the effects, the fourth thing. Gastrin's main effect is secretion of gastric acid. Why? We said it wants to digest food, right? So it secretes gastric acid. This is why, if you remember, it's inhibited by high acidity. Because if you have high acidity, you would not want to increase your gastric acid secretion. So we'll tell gastrin, please stop, okay? The other thing that you also should keep in mind and never forget, it causes hypertrophic effects on the mucosa of the stomach. So it causes overgrowth of the stomach mucosa, okay? And the duodenum and blah, blah, blah. But just know this, because there is a lot of questions about it. Of course, it stimulates motility of the stomach. Why? Because it's, as we said, it wants to digest and empty the stomach from food. Increases insulin glucagon release. This is not very important. And relaxes the pyloric sphincter and contracts the cardiac sphincter. It makes sense when you think that it wants to empty the stomach, so it will relax what? The pyloric sphincter, which is this one, you see here. Yeah. Anyway, so the key point, it's pro-digestion. It wants to digest. And it wants to empty the stomach. And base everything else on that. These are the summary for what we just said. And yeah. Now we have CCK, cholecystokinin. Cholecyst means gallbladder. So its main effect is the gallbladder emptying. What do we mean by gallbladder emptying? Basically, we are causing contraction of the gallbladder itself. And here we have a small sphincter called sphincter of Oddi that prevents the bile from reaching the duodenum when it's secreted from gallbladder. So what will it do? It will relax the sphincter and contract the gallbladder. Very easy. Okay? But as we said, first thing that you should ask yourself when you see a hormone, where is it released from? Eye cells of the duodenum and proximal duodenum. Second thing, what stimulates it? Fat in the duodenum. Important. Remember, CCK is stimulated by fat in the duodenum. By the way, all of these hormones we will use in lecture 5 and 6 and 7. So after lecture 7, inshallah, they will be very easy, okay? So I know it might seem a lot now, but inshallah it will be easy later. It inhibits gastric emptying and helps with intestinal digestion. So it's the opposite for what? Gastrin effect. It inhibits the gastric emptying. So yeah, it's opposite to what? Gastrin. This is also a summary. Most of these you don't need. I think these are enough for you to remember for now. Now, secretin. Secretin, where is it released from? First thing, cells. What is it stimulated by? Second thing, acid in the duodenum and jejunum. Remember this, please. Acid in duodenum and jejunum. And when you see its effect, you will understand why is that. What is it inhibited by? There is no inhibitors here at the moment. There is only one that we will discuss later that inhibits everything. Fatty acids also stimulates secretin. So I want you to remember the acidity is the most important thing for secretin. Why is it the most important thing? Because see what secretin does at the second thing here. It stimulates pancreatic and bile bicarbonate secretion. Bicarbonate is what? A base. So when you have high acidity in the duodenum and jejunum, which are places that you should not have acidity in, okay, they are not made to accept this high acidity, okay, you want to stimulate this bicarbonate secretion to neutralize the acid. So this is why secretin will be released. And this is the most important effect is this. It also inhibits gastrin release and gastric acid secretion. So far, secretin and CCK does, you can say, quite opposite things to gastrin. Okay? Trophic effect on exocrine pancreas, not very important. And yeah, this is what we said. And I also want you guys to keep in mind, as you saw, secretin and CCK, since they are both like quite opposite to gastrin, I want you to know that they both complete each other. So when secretin is released, it's better if you have CCK. And if CCK is released, it's better when you have secretin. So they both work together, okay? Now, gastrinoma. You will find it weird that I put it after secretin, not gastrin. But it's for a reason, you will see. Gastrinoma is tumor of gastrin-releasing cells, which are the cells that start secreting gastrin, okay? And what's also called Zollinger-Ellison syndrome. So what's the problem when you release a lot of gastrin? You will have a lot of HCl, right? What will that cause? Duodenal ulcers, diarrhea, steatorrhea, hypokalemia. Please remember the steatorrhea part. It's asked a lot about, okay? Steatorrhea. Remember it. Okay. Yeah. Now, this is why I put it after secretin. Because gastrinoma tests, to test for gastrinoma, you will do something that we call secretin test, which is the very important part. Secretin, normally, what does it do? What did we say here? Inhibits gastrin release, right? So it decreases gastrin. Now, in these patients who have gastrinoma, you give them synthetic secretin. They already have high amount of gastrin, right? So you give them secretin. What happens? It should decrease it, right? But no. In these patients, exactly for unknown reason, secretin increases gastrin levels. Okay? This is what you should keep in mind. If the serum gastrin goes more than 110 after secretin is given, it's 100% gastrinoma. GIP, glucose dependent insulinotropic peptide. No one remembers the full name, just know it as GIP. Where is it released from? K cells of the duodenum and proximal jejunum. It's stimulated by any type of food, fats, proteins, carbohydrates, whatever it was. Any type of food stimulates the release, okay? But keep in mind, only oral glucose induces it. Only oral glucose induces it. So IV glucose does not. Because it's what released from where? Isn't it the cells of the GIT? So of course, when you give the glucose IV, like through the blood directly, it will not sense that there is glucose in the stomach, for example, or in the GIT, sorry. Anyways, what is its effect? Induces insulin release. I mean, you kind of see it from the name, I guess. And inhibits gastric acid secretion. So far, everything inhibits other than gastrin. They are all working against gastrin. Motilin. Motilin is very different. Where is it released from? M cells of the duodenum and proximal jejunum. Everything is duodenum and proximal jejunum at this point, but yeah, it makes sense. M cells, M for motilin, S cells, S for secretin, G cells, G for gastrin. Yeah, so inshallah it's easy to remember. It's stimulated mainly by nervous control, slightly acid. Yeah, it's not very important to know why is it stimulated, but the most important thing here is the effect. What does motilin do? We call it housekeeping. Basically, when someone is fasting, and remember please, fasting, it's the key word for motilin, okay? When someone is fasting for a while, and they suddenly have, do you guys, for example, when you are a bit hungry, have you ever heard your stomach like moving and you feel like movement of your bowel, and you might even hear an embarrassing sounds and that no one likes and all of that? This is the effect of motilin, because when you haven't eaten for a while, it induces a powerful rhythmic contractions of the stomach, small intestines, everything, whole of GIT, to move everything out. Why? You have been fasting, so there is slightly no movement happening in your GI tract, and bacteria loves things that are not moving, okay? Like water, for example. If water is not moving, it will have bacteria overgrowth. Same thing here. Everything is sitting in the GIT and it's not moving. So this is dangerous to the body. Bacteria will start growing there. So motilin induces powerful rhythmic contractions to get rid of everything when you are fasting. Okay? Now, neurocrines. These were all the classic hormones. Neurocrines, the only important two are these two. From the name neurocrines, they are secreted by neurons. Okay? So, for example, our first is VIP. It's released from parasympathetic ganglia. It has two main effects: relaxation of intestinal smooth muscle and sphincter. Remember when we talked about neurotransmitters in P1? Yeah, this is what it do. It relaxes smooth muscle and sphincters. Very easy. And increases intestinal water. You know, these two, Aslan, we will be talking more about later in other lectures. So, inshallah, it's very easy to remember. Now, this one is very self-explanatory. Gastrin-releasing peptide. Neurokine, so it's released from vagus endings. And from the name, gastrin-releasing peptides. What does it do? Stimulates gastrin release. I mean, يعني. Paracrines. As we said, paracrines, when we have, for example, 5 cells like this, one of these cells will secrete the paracrine and it will affect the neighboring cells. It will not go to the blood, not to the lumen, no, just the neighboring cells, okay? And for our first and most important paracrine, please keep it in mind, somatostatin. Somatostatin basically inhibits everything. If there is acidity, it's released, okay? And it inhibits everything. It's released from these cells of the pancreas. We will see that later again. But I just want you to remember what inhibits everything: gastrin, CCK, secretin. All of these are inhibited by somatostatin. Okay? Histamine basically is similar to gastrin in effect, increases acid secretion. And these guys inhibit it. We will be also talking about that in P5, I think. Clinical case, we said we do not answer clinical cases. Listen, they are important. This one is not. So, what types of cell produce VIB, parasympathetic ganglia cells? Endocrine, neurocrine, paracrine. It's neurocrine. Yeah, you know all of that. So, some questions. Please pause if you want to solve the question. And the answer is C. Here it's asking about increased bowel movement and everything is like moving a lot, so of course it will be motilin. Who increases the motility most? It's motilin, always. Also, so if you wanna solve that, okay, wait a minute, because I think I had, nope, this is very wrong, guys. Okay, sorry, this is E. This is on me, guys, sorry. So basically this question is asking about someone having mild epigastric pain that improves with meals. This means that they have, what do we say? Yeah, they have a lot of gastrin secretion that makes them having high hydrochloric acid even when they are not eating. What does hydrochloric acid do when you do not have food in the stomach? It eats the stomach itself, so it feels like it's hurting them. But when they eat, the hydrochloric acid, instead of eating the stomach, it eats the food. So yeah, this is basically a sign that they have increased gastrin secretion. So the patient's symptoms are most likely due to elevation in a substance with which of the following functions. What does gastrin do out of these things? Promote gastric mucosal growth. We have talked about this. Sorry, I will be editing that in the file. Okay, so cephalic phase of digestion. To understand what is the cephalic phase of digestion, I would like you to know that we divide digestion phases into 3 main phases: cephalic, gastric, and intestinal. We will not be talking about gastric or intestinal for now. So what's the cephalic phase? Have you ever smelled like a very delicious food and feel like your mouth is becoming watery, your stomach is like itching, and you feel you're getting hungry? Yes, this is the cephalic phase of digestion. Okay, and it starts before the food even enters the mouth. It's basically your body preparing to eat, okay, even before eating, and it ends once the food in this is like reaches the stomach, because after it reaches the stomach, it's gastric phase from the name, and after that it's intestinal phase once it reaches the intestines. So from the first stimuli that you have, seeing food, smelling food, whatever, you start the cephalic phase. What do we do during cephalic phase? We produce saliva, gastric acid, pancreatic juices. All over the body is like preparing to accept food. Now, how is this happening? Via nervous control only. And as we said before in P1, nervous control starts very strong above and then it weakens going down. So as you can see, you can even interpret from what's happening here. Okay? But anyways, it's via nervous control and mainly parasympathetic nervous system, and we'll be talking about how is this happening, inshallah, in a minute. So how is this even happening, this cephalic phase? Via what we call conditioned reflexes. You see, this born was, let's take a look at this experiment that was done by a Russian scientist called Pavlov. This dog has an unconditioned response or unconditioned reflex. He was born this way. This is what we mean by that. Okay? Whenever he sees meat or eats, he starts producing saliva. This is how he was born. SubhanAllah. Okay? Okay. Now, each time he was about to eat, this scientist rang a bell. Okay? Which at first doesn't seem like it does anything. But look what happens after it being repeated multiple times. Each time he eats, a bell will be ringed. Each time he eats, once, twice, thrice, 4 times, 5 times. This is repeating for months. And then after a while, what happened? Because of this repetitive pattern that the dog has started to notice, okay, I see food, I hear the bell, or I hear the bell, I see food, or whatever. He's developed what we call this conditioned response, which is that even when there is no meat, just hearing the sound is enough for him to start producing saliva. Can you understand this point? Because now he had developed a connection that whenever he hears the sound, it means there is food. So his body starts preparing. And this is what happened with us throughout our life. We have our food, or our unconditioned reflex, which is the food. We were born this way, which means that each time we eat or see food, For us, it's every time we eat. We start producing saliva and all of that. Now, after a while, our brain developed a connection between seeing the food or smelling the food, because it noticed that each time before we eat, we smelled food. Also, before we eat, we see the food. So it developed this conditioned reflex. And this is what made the cephalic phase possible. Now, only by smelling the food, you feel like your mouth is starting to get watery and all of that. Yes, this is what we call conditioned reflex. Now, the nervous supply of the salivary glands. I want to know that we divide it at first into two: sensory and secretomotor. Sensory, basically. And secretomotor could be parasympathetic or sympathetic, which causes like the secretion of the saliva, and we'll discuss what each of them do. But for now, I want you to know how the innervation of the parasympathetic is happening. First let's start with sensory, Okay, so the sensory innervation, it's gonna be for all three glands that we have: the parotid, the submandibular, and the sublingual, okay, which are our three salivary glands. The three of them will be having their sensory supply by the trigeminal nerve, or cranial nerve number 5. Does that make sense? Okay. What about the parasympathetic innervation? It gets tricky now. Please focus on me. First of all, which nerve gives the supply? The parotid gland. For the parotid gland, as a start, يعني. For the parotid gland, it's the glossopharyngeal nerve. The glossopharyngeal nerve. I want you to remember that we have the pharynx here. Okay? So it's close to the pharynx, so it's basically the glossopharyngeal nerve. Now, what about the other two? They get their supply through the facial nerve. I want you also to remember that they're closer to the face. So they get the supply by the facial nerve. It's just a way to link it, but please remember these two. Okay? Okay. Now, what's the tricky part? I will tell you now. Let's take the parotid as an example. What's happening here is it's getting its innervation from the glossopharyngeal nerve, right? And we said it's a parasympathetic innervation. So there is preganglionic and postganglionic. The fibers of the glossopharyngeal are traveling, and then they synapse onto the ganglion. But wait, which ganglion is that again? Yes, the trigeminal nerve. So it synapses onto the trigeminal nerve itself, a branch of the trigeminal nerve. Does that make sense? And once it synapses on that, it sends its fiber through the trigeminal nerve branch and to the parotid gland. Wait, does that mean that the postganglionic parasympathetic here is the trigeminal nerve? No, it just works as a pathway. So you can think of it as if the glossopharyngeal came here and said, okay, trigeminal nerve, you're gonna go anyways, right, to the gland, because you're given the sensory innervation, right? You're gonna go anyways. Why don't you take me with you? Does that make sense? So it sends its fibers through the branch of the trigeminal nerve. And so does the facial nerve. Same idea, but with the lingual and sublingual, and submandibular glands. It synapses and it sends its fiber through the trigeminal nerve. So if a question comes, which of the following gives the parasympathetic innervation for the parotid gland? Never say trigeminal. Okay? It's going to be the glossopharyngeal, as we discussed. But it travels through the trigeminal nerve. Now, I want you to remember something. And yeah, it's here. Two things for each. Just two more informations that I want you to know. The ganglion that the glossopharyngeal synapses onto that we discussed is called the otic ganglion. Please remember. Otic from ear in Latin. And parotid is like next to the ear. So it makes sense. And then the branch of the trigeminal nerve that will take the glossopharyngeal nerve fibers with it is called the auriculotemporal nerve. So it's coming from the glossopharyngeal to the otic ganglion, and then the fibers of the glossopharyngeal are traveling through the auriculotemporal nerve. Temporal, sorry. Now for the submandibular and sublingual glands, as we said, facial nerve, synapses, and submandibular ganglion. You can think of it submandibular. Yeah, makes sense. Yeah. Travels via the lingual nerve of the trigeminal nerve. Also lingual nerve, sublingual gland. Yani, try to link it. And as we said, no parasympathetic innervation from the trigeminal nerve. It's just a pathway, a car, a ride, whatever you want to call it. Saliva secretion. So we are now going to secrete the saliva. But what's happening to the saliva? It goes into two major stages. We produce it in the acini and then modify it in the ducts. And you will see this pattern a lot, inshallah, throughout the physiology of the GIT. We secrete in the acini and we modify in the ducts. So, the first stage is in the acini. It produces an isotonic plasma-like solution which is rich in salt. I want you to remember, it's isotonic and rich in salt. This is the saliva that got released from the acini. Now I want to ask you, it's rich in salt. Is your saliva salty? No. So what happened in the ducts? Can you interpret that? Yes, we reabsorbed the salt in the ducts. So this is the second stage, which we reabsorb the salt. And in exchange, we have to release something or secrete something, and it will be bicarbonate and potassium. Bicarbonate and potassium are secreted. Does that make sense? Okay. Now we reabsorb the salt. Does that mean the water will be reabsorbed? Water full of salt. No, because the ductal cells are impermeable to water. So even though the salt was reabsorbed, the water is staying there. What does that happen? What does that cause to the hypotonicity of it? To the tonicity of the saliva. It made it hypotonic. Once it reaches this hypotonic stage, it's always hypotonic under all circumstances. It will not go back to being isotonic or hypertonic or whatever. It might be less hypotonic, more hypotonic, but it's hypotonic. The only place where it is isotonic is in the acini before it goes to the ducts. How does aldosterone affect it? Same way it affects urine. Increases sodium reabsorption, increases potassium secretion. Very easy to remember. And please know that aldosterone effect is universal. So if there is cells that aldosterone affected, it will have this effect always. Now, why and how would saliva composition change in a patient who has been taking diuretics for a month? Knowing the last information that we said. Well, he had been taking diuretics, so he will be losing a lot of volume, which activates the renin-angiotensin-aldosterone system. We have aldosterone in here, right? What did aldosterone do? Exactly. Absorb sodium, secrete potassium. Now, this is very important concept to grasp on, which is the flow rate of the saliva and the ionic composition. Now, when you increase the flow rate, which is, by the way, happens mostly by parasympathetic activity, rest and digest. What we mean by increase the flow rate is that it will be flowing through the ducts very fast. So that leaves the cells with less time to modify it. So for example, if it had 10 seconds to modify the saliva before it was secreted, now it has 2 seconds because it's moving fast. Does that make sense? Okay. And what does that lead to? Makes sense. Sodium will increase by increasing the flow rate. Why? Because it was initially reabsorbed, right, in the ducts. Now the ducts cannot reabsorb it well because it's moving too fast. So the concentration increases. So does chloride. And potassium also makes sense because it was initially secreted. So it will drop because there is no time to secrete it because the flow rate is increasing. The one that doesn't make sense here is bicarbonate. Bicarbonate is secreted. So we shouldn't have enough time to secrete it when we increase the flow rate, right? Why is the concentration increasing? Because of parasympathetic nervous system activity. Basically, we cannot afford to make saliva without bicarbonate. Bicarbonate has to be in the saliva. It's the major thing there. So how does our body accommodate the high flow rate? It tells the parasympathetic nervous system to start stimulating bicarbonate secretion specifically. So it adapts. So just to remember, all of the three make sense. You can interpret it, inshallah. Bicarbonate, just remember that it's the exception. So it increases. We can't afford saliva without bicarbonate. Now, some notes to keep in mind. Fear, stress, depression, or anything that inhibits parasympathetic activity or stimuli or whatever or anything that blocks the parasympathetic activity, such as the atropine that we will be discussing in a minute. It decreases the flow rate of saliva and bicarbonate secretion. Makes sense. Parasympathetic nervous system affects the saliva. How? Increasing water, making it more watery, more ions, enzymes rich, and all of that. And also, there's this side effect, or secondary effect. It dilates the vessels supplying the glands. And this does what? Increases blood flow to the glands, and hence increases filtration, and basically it will work more. Now for the sympathetic nervous system, it does not inhibit. Please do remove this misunderstanding now. People think that parasympathetic activates, sympathetic inhibits. It does not inhibit directly. Sympathetic actually stimulates, but it stimulates a different type of secretion, which reduces the overall volume. But what does it secrete? Thick, viscous saliva that's filled with enzymes. Please remember this. So it does not inhibit. And this is a summary. I just want to note a few things here. These things are important to know because there had been questions about it before in the past, and not from Dr. Angelica, I think, but there had been questions. And I want you to note here, we have the dehydration, fear, sleep, whatever the things that we discussed, they inhibit parasympathetic activity and hence inhibit saliva. These things activate parasympathetic activity and hence activate saliva. So just please remember them. And as you can see, we have also here atropine. I want to talk about atropine. Atropine is a plant or a chemical coming from a plant which blocks this muscarinic blocker of muscarinic receptor or M receptor. Sorry, not muscarinic. Sorry guys, please. M cholinergic receptors. This is M cholinergic receptors. You can see the M. So atropine blocks it. And hence the acetylcholine will not be able to bind to the M cholinergic receptors, and it will not produce the parasympathetic effect. So it's basically a parasympathetic inhibitor, or specifically for acetylcholine. Okay, I'm sorry for the mistake. Now, the CFTR proteins. CFTR proteins are channels for chloride. Okay, they secrete and reabsorb chloride depending where, but they mostly secrete. Okay, let's say that they mostly secrete. And water will follow where possible. So, now think of me what will happen when it's impaired. In the acini, the chloride ion and bicarbonate ions are not secreted into the saliva. Now you will say, wait, chloride is reabsorbed, right? Yes, but in the ductal cells. Here it's in the acini. How did we get the chloride at the first place, aslan, to reabsorb it in ductal cells? Yes, by secreting it into the acini. Or in the acini, yeah. So this secretion will be impaired, both chloride, bicarbonate, and water, because water will not be able to follow the ions. They are not secreted, aslan. In the ductal cells, there will be, of course, decreased reabsorption of chloride, as we said, and hence sodium, because sodium follows chloride, whatever. At least here, yeah. And water will not be affected. Why? Yes, because the ductal cells are impermeable to water. So water cannot follow anyway, so it doesn't care. Yeah. Final saliva will be viscous because we had less water, concentrated, less water, acidic because we failed to secrete bicarbonate into the saliva. Now, saliva functions, they are very general stuff that you don't need to remember specifically. But I want you just to focus with me on the digestion part, because we will see how it helps with the digestion. Now, there's two main enzymes in saliva that are important in chemical digestion. And the first one is alpha amylase. Alpha amylase is the one that starts carbohydrate digestion in the oral cavity. And even after it reaches the stomach, it works for a little bit. But remember, it's an enzyme, protein. So it gets denatured by gastric acid. So its effect is mild. Now for lingual lipase, this does not function in the oral cavity. It's produced in the saliva, but it cannot function. Why? pH. It needs an acidic pH to work on. And you remember the saliva had high bicarbonate, okay? So it cannot function there. So what happens? Once it reaches the stomach, it finds the optimal pH that it's looking for. And then in the stomach, it starts working. And it's not denatured. It can overcome the denaturation that kills the alpha amylase, so it can work even better. And yeah, it's present under high sympathetic activity. You can remember that. But as we said, also sympathetic gives us a thick, full of enzymes thing. Oops, what's this? Okay. This is just a summary for all of what we said. And yeah, I think nothing new here. Please pause if you want to solve it. Here are the answers. The answers. As I said, cholinergic receptors, okay? Please remember that. I don't know why I said muscarinic. Okay. And yeah, 4 questions. Please pause if you want to solve it also. And it's B. Now, we said we increased the flow rate, right? So this and this are gone. Because when you increase the flow rate, what happens to Na or sodium? The concentration increases. And for the potassium, it will be, cell B, C, D, one of the 3. So this is what we will be judging based on. Does the saliva become isotonic again? Never. Hypertonic again? Never. So it can't be anything but B. It remains hypotonic, sodium increased, potassium decreases. The answer is B. And yeah, it's basically as we said, parasympathetic is using cholinergic receptors to start with, so anything adrenergic is gone aslan. And its concentration or dilation, we said via dilation. And yeah, so just by this you can interpret it and indicate the case of enzymes, amount of saliva, blah blah blah. Now something that I want to note in the last question here, the increase in flow rate. If you I will go back to it inshallah after we finish the last question. Of course, it will be the impermeability of ducts for water. But then something that I didn't mention in the flow rate, and I think it's a bit important to know, that you can interpret it yourself, but I will help you with it. We said it does not become isotonic again after it becomes hypotonic, right? Okay. But what happens to the tonicity when the flow rate increases? Of course, it will remain hypotonic, but like more hypotonic, less hypotonic. What happens? It becomes less hypotonic, so it is approaching plasma values, as you can see, because sodium is increasing, chloride is increasing, almost everything is increasing except for potassium. So it's getting closer to reaching the isotonicity of the plasma. But of course, it fails to do that because خلاص, we're done. Once it's hypotonic forever. And yeah. Okay, esophageal motility. First, let's start with the nerve innervation of the esophagus. To understand it, we need to divide the esophagus into 3 thirds. Upper third, middle third, and lower third. Upper third, closest to the mouth, will be skeletal muscle. Makes sense. Lower third, closest to the stomach, will be smooth muscle. Makes sense. The middle third will be mixed. A little bit of this, a little bit of that. Easy. The nerve innervation depends on the kind of muscle we're talking about. If it's skeletal muscle, it will be innervated by somatomotor branches of vagus. Because vagus does not only give parasympathetic innervation, it also gives some somatomotor branches for skeletal muscles of the esophagus. Easy. We said the enteric nervous system is present from the start of the esophagus until the end of the anus, right? So what's going on here? Do we have it here? But this is in smooth muscle, so what's happening? It's present, but here in this part, it has sensory functions only. So it senses, but it doesn't do peristalsis because this is not smooth muscle. Smooth muscle, on the other hand, is also innervated by autonomic nervous system, the same boring concept that we already know. Parasympathetic by vagus and sympathetic by T5 to T9 segments of the spinal cord. I don't think she will ask about the levels, but yeah, I just put it for the sake of, complete information. Now, the parasympathetic nervous system innervation of the esophagus and the lower esophageal sphincter. The preganglionic fibers, the parasympathetic fibers, if you remember from B1, where do they synapse on? On the enteric nervous system itself, right? Because the enteric nervous system gives the postganglionic fibers, if you remember that. if they synapse on the enteric nervous system, which plexus exactly? It's our Bach's plexus. Our Bach's plexus. Synapses on what? Our Bach's plexus. Okay. Both ganglionic parasympathetic nerves are coming from our Bach's plexus, and it innervates the muscles. If we are in the lower esophageal sphincter, and this is the main point that I wanna say, you want it to relax it or to constrict it, like when there is food. Of course, you want to relax it, right? Because it's usually constricted by itself. So it will release what? VIB and nitric oxide to relax the sphincter. It's a concept that we have discussed even in P1. Now the swallowing process. What's happening while you're swallowing is that there is a voluntary part that you have control over, and there is involuntary part that you do not have control over. Okay? While the food bolus is still in your mouth, you have control. Okay? So what happens here when you want to swallow? You see your tongue pushes the food back, and the nasopharynx closes. Why would the nasopharynx close? Just imagine here, for example, okay? The tongue pushed the food back. I would want blue color here. Okay, the tongue pushed the food back, but still the food can go up to the nose. So this nasopharynx will be sealed by what? Contraction of the soft palate. Okay. So now where can the food go? Only down and only down from here. So what when it goes down, what options does it have? It can either go to the esophagus or to the pharynx and into the esophagus, or it can enter the larynx, and that's a big no. We don't want it to do that. So what will happen? The larynx will elevate. Back to red. Okay. The larynx will elevate. The airway will be closed. The upper esophageal sphincter opens to accept the food. Easy. Very easy concept, okay? You do not need to memorize each step and the seconds, and just have the idea. Again, this is just a summary. Tongue pushes food back. We don't want it to go to the nasal cavity. So soft palate closes the nasopharynx. It can only go down now. Pharynx or larynx? We want it to the pharynx. So we will close the larynx, elevate the larynx, and close the epiglottis. Close the epiglottis. There was a question about this in the 17th batch one. Our esophageal sphincter will relax, and the pharynx contracts to push the food down. Food reaches the esophagus. Very easy. Now, questions also that was found in Dr. Angelica's slides is this note. Not slides, sorry. The practice questions. And it's that the palatopharyngeal folds are responsible for determining whether or not can a bolus pass to the pharynx. So the palatopharyngeal folds are basically like an arch that's found at the back of your mouth. And it's responsible, if it's too big, it will not pass them. Just know them because there was for some reason, there was a box question about this. Okay, esophagus parts and function. Easy. Upper esophageal sphincter. What's the main idea? Okay, it protects the airway from swallowed material because you don't want the swallowed material to go back up. And it protects the airway from gastric reflux. So if someone has GERD and the acid is going up, this sphincter will act like a, as a protective effect. The middle part of the esophagus, it pushes food down and all of that. I mean, these things are actually very self-explanatory. I don't know why I'm doing this. But anyways, the lower esophageal sphincter is also to protect the food from coming back to the esophagus and so on. Now we want to talk about peristalsis. We have discussed it in the P1, so it's very easy. The contraction before the bolus and relaxation after the bolus. So it will move this way. And once it reaches here, it happens again, contraction before, relaxation after, through the enteric nervous system that we have discussed. But here in the esophagus, there is something that you should keep in mind. Is that peristalsis doesn't happen in one wave. It's two waves of peristalsis, primary and secondary, and this is important. Primary peristalsis is initiated by swallowing only, and it's mediated by vagus. So, once you swallow the food, the vagus nerve senses that, and okay, خلاص, we will start peristalsis. Okay? Very easy. Secondary peristalsis happens when primary peristalsis fails to clear the esophagus. Have you ever eaten something and you didn't chew it well, and you felt like it's stuck for a second in your chest, and then you feel it has been pushed down again. This is when the primary peristalsis failed, and what this second push down is the secondary peristalsis. And who does it? Enteric nervous system. You remember when it senses the stretch and it does blah 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 peristalsis? Yeah, this is the secondary peristalsis. Esophageal manometry. It's a recording of the pressure inside the esophagus. So as you can see, when we look at, for example, the upper esophageal sphincter, here, this, you will see that it's normally constricted. So what will be the pressure? In other parts of the esophagus, it's 0 at rest. But for the sphincter, even at rest, look at how much the pressure is. 60, can you imagine? And then here is the swallowing part. Let's take the blue for this, okay. So it's 60, and then the swallowing part, it relaxes, because you just swallow. So once it relaxes, the pressure drops down, and then it contracts again, and it continues after the swallowing. Very easy. Throughout the whole esophagus, it makes sense what's going on. It's usually 0. When the food is here, the pressure increases because it contracts, and then it goes down, down, down. Here's the part also that you should keep in mind is the lower esophageal sphincter. It also has what? Higher pressure than 0, but it's less than the upper one. And once you swallow, the pressure drops and then it goes back up. And yeah, just to read that, it's same as the upper esophageal sphincter. Why are we discussing that? Because you will see in diseases, this esophageal manometry that makes sense becomes very weird, okay? And you can diagnose diseases from that. What are our diseases that we will discuss? First, let's start with achalagia. Achalagia means failure to relax. It's a disease where the lower esophageal sphincter fails to relax. So the food is accumulating before it and it's being stored in the esophagus, but it doesn't pass to the stomach because it's not relaxing. Okay? So what do you think will happen when you look at the manometry for those patients? Of course, the pressure of the lower esophageal sphincter will be increased because it's constricted. Okay? And this is most likely caused by loss of enteric nervous system in the lower esophageal sphincter. Do you remember Hirschsprung's disease? It's the same idea. Same idea. You don't have inhibitory neurons, so it's not relaxing. Very easy. Now, something also that you should know is that the stomach naturally has a reflex that's called receptive relaxation. This basically, the last part here, or the start of the stomach, relaxes when it senses that food is coming, to allow food passage without like any extra pressure or any extra resistance, okay? This receptive relaxation is lost here in achalagia, okay? This is what you see when you have barium swallow, bird's beak. Can you see the resemblance? This is the, it's as the same idea. See the bird's beak? So this is the esophagus, by the way, and it's filled with food, but at the lower esophageal sphincter, it's constricted. Similar to Hirschsprung, if you remember. Okay, so what happens in the esophageal manometry when you look at these patients? You'll see that the resting, this is the normal esophageal manometry. The resting lower esophageal sphincter is, for example, let's say 20. And then when you swallow, it decreases to 0, and then it increases again. Now what happens in these guys? It's almost 60 or something. And when you swallow, it doesn't even go down much. Just a little bit, and then it goes up. So, it's, this is what's happening. GERD is the opposite to achalagia. There, the lower esophageal sphincter cannot relax. GERD is the lower esophageal sphincter does not close well. And what would that lead to? The stomach acid going back or refluxing to the esophagus. And this might cause heartburn and blah, blah, blah. Clinical case. What's primary esophageal peristalsis initiated by? Swallowing. Secondary, esophageal stretching and enteric nervous system mediated by enteric nervous system. How is gastric reflux prevented? Closure of the lower esophageal sphincter. Questions? Okay, if you want to answer it yourself, so. And it's B, because you have bird beak appearance, so this is achalagia. What happens in achalagia? Failure of lower esophageal sphincter to relax during swallow. Very easy. Now, Okay, it's C. And basically, the food is coming back to the pharynx, and this impairs this function, preventing respiration of esophageal contents into the airway, because if it reaches the pharynx, it will go into the airways. The others doesn't even make sense, so yeah. Now, gastric response to a meal. Very easy lecture, and it's very dependent on P2. So please, if you don't understand P2 well, go back to that. No, but inshallah, no, don't go back. Just come here. Inshallah, we'll deal with it. Let's divide the stomach into 3 parts to understand this concept that I will be talking about. You have the lower esophageal sphincter and cardia. I know, lower esophageal sphincter is part of the esophagus. That's not something that we'll discuss here. It's just that they have similar functions, so we will just put them together. Now we have also the fundus and the body. This is the fundus and the body, the yellow part here, and antrum and pylorus, this part, which we mix food here and grind it and all of that. We'll discuss more later. Why did I divide it this way? Because you will see secretions in the stomach is different depending on different parts, or on which part are we talking about. So when we are in the lower esophageal sphincter and cardia, the first part, it's mucus and bicarbonate. In the antrum and pyloric, it's the same. So if you're at the start or at the end, you're secreting mucus and bicarbonate. Bas, done. Now, the fundus and the body, they also secrete mucus and bicarbonate, because you cannot afford to lose that function, mucus and bicarbonate secretion. But we will add a few things. H+, intrinsic factor, pepsinogen, lipase. Don't memorize for now, because we'll discuss them more one by one now. Now, gastric gland cells, very, very, very important. These glands, either they are in the body and fundus or in the antrum. This classification is not very important, but just so you have it in mind. We have parietal cells. Parietal cells secrete hydrochloric acid and intrinsic factor. Intrinsic factor is very important for B12 absorption that happens in the ileum. So what's going on? We're releasing intrinsic factor from the stomach. B12 attaches to it and it gets absorbed in the ileum. Easy. And of course, the main function of parietal cells is hydrochloric acid secretion. Chief cells secrete pepsinogen and lipase, which are both enzymes that we will talk about later, but pepsinogen is the most important one. So we know hydrochloric acid for parietal, chief pepsinogen. ECL cells, I will not read that long name, releases histamine. And we'll discuss histamine function in a minute. In the antrum, G cells secrete gastrin. Do you remember our friend gastrin? Very easy, I think you already know that. And we have mucus neck cells to secrete mucus and bicarbonate, but that should be everywhere, but the doctor put it in the antrum, so whatever. This is a summary for each cell, what does it secrete, the function, blah, blah, blah. I will not go through all of that, but because we will take it one by one, inshallah. But just something that was not what was not mentioned before is the D cells, somatostatin. We mentioned it in the P2, but still, we will talk about it here. So, I know this sounds scary, sounds like a lot of things are happening at once, but it's very easy. I want you to focus with me. We have phases of digestion, right? In each phase, there will be certain things that are released to do certain functions. Okay, do you agree on that? Okay, let's see what happens. And our end point that we want to reach is, do we increase hydrochloric acid secretion or do we inhibit? This is what we will discuss in this lecture. So let's start with the cephalic phase. You are waiting for the food to come. So you will stimulate hydrochloric acid secretion. Makes sense. So it stimulates hydrochloric acid secretion via vagus control. Either acetylcholine directly to parietal cells or stimulation of gland cells by indirect pathways. And we will discuss the parietal cell in a moment. Now, when we go to the gastric phase, the food is here. So you want to digest it. So you will stimulate HCl secretion through nervous control and hormonal control. Nervous control by enteric nervous system, which is Meissner's plexus, if you guys remember, that increases secretion, or by vagus control. Hormonal by gastrin and histamine. Very easy, very easy. You already know that gastrin and histamine stimulate hydrochloric acid secretion. Now there is, when you have high acidity, what happens to this phase? It's inhibited. Why? Because you already have high acidity. You want to increase HCl, you will eat the stomach out. You do not want that to happen. And also proteins stimulate. You know, gastrin, we talked about the stimulus and all of that in P2. Phases of digestion, last one, intestinal phase. we are done using hydrochloric acid. So what are we doing? Mostly inhibiting hydrochloric acid secretion via multiple hormones. Somatostatin, secretin, GIP, I will not read that name, glucose, okay, I will not. CCK, cholecystokinin, all of these guys inhibit hydrochloric acid secretion, okay? But out of them, I want you to remember that somatostatin is the most associated with the decreased hydrochloric acid. Why? Because it's the most direct one. We will discuss that in a minute. Cholecystokinin is most associated with decreased gastric emptying. And it's stimulated by fat, acidity, hyperosmolarity of chyme, blah, blah, blah. It's not cholecystokinin only when I mean stimulated by fat. It's the whole phase. When you have high acidity, high fat amount, all of that, it will simulate this intestinal phase. So when you look here, now it makes sense. When you are talking about cephalic phase, you are going through the vagus nerve to histamine, acetylcholine, gastrin, blah, 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 all of these guys to increase hydrochloric acid secretion. When you're in the gastric phase, it's mostly gastrin, still increasing hydrochloric acid secretion. Intestinal phase, it's mostly going through these guys, which release what? Inhibiting hormones. Okay, question. Vagotomy abolishes cephalic phase of gastric secretion. Why? We said the cephalic phase is only by vagus. Vagus is the one who controls everything in cephalic phase. So if you don't have vagus, you don't have cephalic phase. But here, when you block, when you use a muscarinic blocker like atropine, you do not abolish the cephalic phase fully. Why? Because vagus does not only release acetylcholine. Muscarinic blocker blocks what? M-cholinergic receptors, which are receptors for acetylcholine. But there are other things that vagus still can secrete in the cephalic phase, such as gastrin-releasing peptides, if you guys remember, which also releases gastrin, basically. So it will not be fully abolished. Very easy concept, but I don't know like if she will ask about it aslan or not. Hydrochloric acid secretion by parietal cells. Okay, what's happening in the parietal cells? Now, in order to make hydrochloric acid, we need to think hydrogen and chloride. Where do we get those? Please focus with me. These cells have a lot of mitochondria. Okay, why? Because they use a lot of ATP. So they have a lot of mitochondria, hence they have a lot of carbon dioxide. When they have carbon dioxide with water, okay, this reaction that we memorized 3,000 times, okay, I hope you already know it. It gives us hydrogen and bicarbonate. Easy, easy. So we have got our beautiful hydrogen. So we bump it out. How? By this important bump. We'll have a question about this, okay? So please remember the bump. Hydrogen, potassium, ET base. So we had got our H plus out. We just need now the chloride. How? Remember when we weighed here hydrogen and bicarbonate? We can use that bicarbonate to get our chloride. How? From the blood. Chloride is coming from the blood. Chloride. Once it reaches here, it will be exchanged for bicarbonate. And we throw the bicarbonate to the blood. Now we have chloride here. It will go through channels. And now we have got our beautiful chloride. So now we have our H and our Cl. Very easy. And please, I've seen a lot of past questions about this. I didn't put a star because it's not from Dr. Angelica, but the concept itself is important. Parietal cells activity control. We have talked a lot about parietal cells and that they secrete HCl, but what activates them and what affects them directly. This is just a summary for everything. This is what you need, to the point. Stimulated directly by gastrin, histamine, acetylcholine. Inhibited directly by somatostatin and prostaglandins. You already know these three, okay? And what they do, supposedly. And you know that somatostatin inhibits. The only new one is prostaglandins. Just know that it inhibits also. Okay. And know that histamine is blocked by cimetidine. And acetylcholine is blocked by atropine. You know that already. Muscarinic. This note is very important also. In patients with gastric ulcer, omeprazole is used to relieve them since it inhibits hydrogen, potassium, ATPase. Can you see that? Omeprazole inhibits this pump, and it's the most effective way of decreasing hydrochloric acid secretion. Summary, you already know that. Intestinal phase, we have talked about it. It's stimulated either by high acidity or high tonicity or solutions or blah blah blah. It secretes 3,000 things with the somatostatin, secretin, GIP. I forget always what it's called. CCK. All of these guys, what do? Inhibit parietal cells, either directly or indirectly. The thing I want to add here is these long and short reflexes. We know short reflexes and long reflexes, but what's special about intestinal phase is that long reflexes are not parasympathetic pathway. They are sympathetic. Because why? You want to inhibit, right? Who inhibits in the GI tract? Sympathetic or parasympathetic, generally speaking? Sympathetic. Because, you know, parasympathetic is rest and digest, like it's from in the name, literally, okay? So it's rest and digest. So here, the long reflexes are happening by a sympathetic pathway. Questions? Which of the following factors have a direct action on the parietal cells to stimulate acid secretion? It's C. You know, gastrin, acetylcholine, and histamine. They all stimulate secretion. Somatostatin is a no because it inhibits. Very easy. To medical students, well, you can stop if you want to read it. Okay, I'll just move on. It's gastrin-releasing peptides because which of the following basically increases HCl or whatever. It will be gastrin-releasing peptides. Okay. By the way, this is VIP because I don't remember if I told you the full name or anything. But when you see it, I think you can tell it's VIP. But it's not even the correct answer anyway, so yeah. 6. Quite a long lecture, this one, so like fasten your seats. Hydrochloric acid functions, a lot of unnecessary stuff. Just know the pepsinogen activation is the most important one. Okay? Remember, pepsinogen is released from? Yes, chief cells. Okay, so chief cells secrete pepsin in its inactive form. Because if it was released in its active form, it would have eaten the cells themselves, right? Because what does pepsin do? It breaks down proteins. And if it's inside the cell and it's activated, it will break down proteins inside the cell, which is a big no-no, okay? So we release it in its inactive form. To activate it, we need a pH of 3 to 5. What provides us this pH? Hydrochloric acid, exactly. So parietal cells help the pepsinogen activation. Okay. Now we have pH of 3 to 5. It starts activating. Once it reaches below 3, it starts rapidly activating, like as a cascade, very quickly. Okay? When you activate it, you need to maintain a pH of 3.5 or less to have it activated. Either other ways, it will start deactivating again. This is why when there is no food or anything, the pH starts increasing. This is a protective mechanism where it becomes inactive again. Now we have protective versus damaging factors of the stomach lining. The protective factors are, of course, bicarbonate, mucus, prostaglandins, because they inhibit parietal cells, if you remember, tight junctions between the cells, and growth factors. Okay, growth factors because they replace the stomach lining every 3 days, I think. Can you imagine? Like a whole set of cells is replaced. Damaging factors. And by the way, this is why cancer is very common in the stomach, because the cells are, okay, you know what I mean. Now, damaging factors, you have H+ and pepsin, NSAIDs and H. pylori. We will talk more about them, but the first one is self-explanatory. The rest we will be talking about now. For H. pylori, it's a bacteria. What does it do? It causes peptic ulcers. How? It inhibits when you have an infection, okay, of H. pylori, it inhibits somatostatin. So that increases gastrin. And that gastrin, while it increases H+ secretion. So it leads to duodenal ulcers. Same thing, it also decreases bicarbonate secretion and blah, blah, blah. So basically, what I want you to keep in mind when you think about protective versus damaging factors is that if these guys go down and these go up, you will have an ulcer. Okay? Makes sense? So this is going down, this is going down, and these are going up. So of course it causes ulcers. Now, here's something that you should keep in mind. How does H. pylori maintain itself protected from the stomach acid? Like, it's a bacteria. Shouldn't the stomach acid dissolve it? No. It has this enzyme called urease. Turns urea into ammonium. Ammonium is very basic. So it protects the bacteria. And at the same time, it's one of the main ways these bacteria are damaging the stomach is by ammonium also. So ammonium not only protects the bacteria, it damages the stomach mucosal lining directly, because it's very basic. So, to summarize, gastric and duodenal ulcers. Duodenal ulcers are mainly by excess H plus secretion. Why? Because it's not made to withstand this amount of acidity. The duodenum, I mean. So it will be mainly by H plus secretion. But for gastric ulcers, they can maintain themselves and protect themselves against the high acidity. But the main reason is defective mucosal barrier. So if the tight junctions are gone, if there is less protective factors, this leads more to stomach ulcers. Okay, if someone takes NSAIDs, they will suffer from peptic ulcer disease. Why? This is something that I forgot to mention, but NSAIDs directly inhibit prostaglandins. So they decrease the protective factors. And we said if you decrease protective factors, ulcer. Increase damaging factors, ulcers. Now, why would we give antibiotic for someone with peptic ulcer? Because if it's H. pylori, it's a bacteria at the end of the day. Antibiotic is enough to kill it. Sphincters of the GI tract. They are closed when inhibitory neurons are inactive, open when inhibitory neurons are active. Very self-explanatory. If the inhibitory neuron, which releases VIP, is active, it will relax. If it's inactive, it will close. I mean, self-explanatory. Now, slow waves and action potentials. This is a very important concept. Slow waves, what are slow waves? They are waves of depolarization that are happening spontaneously in the GI tract. So the cells of the GI tract have this trait where they can generate their own slow waves or action potential. But what's the difference? By the way, this is not how you write potential. It should be with one T. So these slow waves, what do? They are waves of depolarization that are very close to the threshold, but they do not usually reach the threshold. What do I mean by that? Action potential is an all or nothing response. Okay? So if you reach the threshold, an action potential is generated. If you do not reach the threshold, you do not have action potential and no muscle contraction at all. So if the slow waves manage to reach the threshold, it causes an action potential. But it doesn't always succeed. Is that idea like easy to understand? Okay. What generates these slow waves? Cells in the GIT smooth muscle, in the GIT interstitium called interstitial cells of Kajal. This was a question. Okay, there was a question about this. Interstitial cells of Kajal. They are what generates slow waves. Now, to understand slow waves and action potentials, let's look at this. Okay, so you have this membrane potential, which is at resting membrane potential, and then slow waves start increasing, and it goes down. If it manages to give us one action potential at least, you will see this spike potential that we call. Okay, and you see the muscle tone has increased because it managed to give us one action potential. And then it tries again. This time it managed to give us two action potentials. So the muscle tone will increase. And sometimes it gives us one, but the muscle tone is still not that much. This is at rest. What if it's stimulated by parasympathetic pathways? You will see that one slow wave manages to give us what? 4 action potentials. A lot of action potentials are generated from slow waves. So they reach the threshold more often. And that causes the muscle tone to be very high compared to the initial. What if you inhibited via sympathetic pathways? You will see slow waves do not manage to give us any action potential at all. And the muscle tone is dead. Same thing here. This slow wave approaches the threshold but failed. This one managed to give us action potential. This one, 2 action potentials. If you stimulate it, you will see 300 action potentials or something. And yeah, this is the concept. Now, phasic versus tonic contraction. The difference is phasic comes at phases. So you sustain it for a few seconds and then it goes away. Similar to this is peristalsis. Also, like an easier even concept to understand, try to flex your biceps. You see it contracts and then goes away, right away. So yeah, you cannot sustain it for too long. Tonic contraction, on the other hand, is sustained for hours. And this is only found in the sphincters and anterior portion of the stomach. And we will discuss that now. You will see why is that important to have in the anterior portion of the stomach. Adaptive relaxation. Adaptive relaxation is a mechanism that the stomach have. Do you remember the receptive relaxation? It's similar, but this time it's for the whole stomach, where it basically relaxes the whole of the stomach and to withstand the high amount of food. Okay? Because you don't want the stomach to be very contracted when you have high amount of food. So it relaxes to allow food to come in and doesn't change the intergastric pressure much. How does it happen? Vago-vagal reflexes. So vagus takes the signal. Can we go back to red, please? Okay. Takes the signal to the brain, and then vagus takes the signal back from the brain, and it tells the enteric nervous system, relax. And this is what happens. Here, it's adaptive relaxation. Here, it's tonic contraction. This. And this is what the stomach is always doing. Subhanallah, it has this mechanism. Either it contracts for hours. Do you remember when I said the tonic contraction is in the anterior portion of the stomach? Because can you see this line? This is the normal stomach. If it needs to decrease its volume because no food is coming or anything, it can maintain this tonic contraction to decrease its volume. If it's very used to having a high amount of food, it can do by adaptive relaxation to maintain this. So it's moving between tonic contraction and adaptive relaxation. It can either relax or contract for hours. And this is why the stomach is called a reservoir for food. Because food is kept there. It's not merely for digestion, by the way, the stomach. Most digestion is happening in the intestines anyways. But it carries the food and it adapts to the amount of food that you eat. This is why some people might have a big stomach and some people might not have a big stomach. Like actually, if you saw the cadavers, you'll see that some of it is like a small bag and some of it is like, a giant bag. Whatever. But you'll get the idea. Now the mixing in the stomach is happening in a retrobulging mixing way of mixing. And what do we mean by retrobulging mixing? Basically, the stomach contracts from up to down, up to down, up to down, until the food reaches the pyloric sphincter, down. Once it reaches the pyloric sphincter, the antrum will contract the food and pushes it towards the pyloric sphincter. If the pylorus is closed, what happens? It will face like a wall. The food will face like a wall, and it will hit the wall and go back up. So it will strike and go back up. And then it starts contracting again and again and again, and it goes in a cycle. So we are contracting from up to down. If the pylorus sphincter is closed, it hits it and goes back. And do the cycle again, repeat the cycle. And this cycle is repeated each 3 minutes. Is that easy? Okay. Now this is only if the food is not digested well and it's not yet ready to move to the duodenum. But what's happening after some parts of the food can pass through the pyloric sphincter? Once it goes to the duodenum, now the duodenum works as a sensor. It senses the chemical properties of the chyme. If it's hyperosmolar, high in fat, acidity. Do you remember what starts the intestinal phase when we discussed in P5? All of these things. If the duodenum senses it, it releases these hormones like secretin and all of that to close the sphincter. Because no, it's not ready yet. Because, look, the pyloric sphincter is not only part of the stomach. It's shared between the stomach and the duodenum. So the duodenum can also affect it. So if it's ready to move, the duodenum will tell the pyloric sphincter to relax a little bit. If it senses that no, it's still high in acid, I cannot take it yet, it tells the pyloric sphincter to maintain itself closed. And so on and so forth until the food is ready to pass. Easy? Easy. Of course, nervous and hormonal control are what's affecting the sphincter. Okay, now for solids versus liquids. This lecture is so long. Anyways, solids versus liquids. It's very self-explanatory. I don't even know why is that a part of the lecture, but okay, whatever. If the food is solid, it will not pass very easily. If it's liquid, it will pass easier and faster through the stomach. And you can see here. This is a liquid meal. Once it goes into the stomach, it will start going out. We have this lag phase here. This lag phase is the phase where 0% of the food that was like got into the stomach is fit enough to move through the pyloric sphincter yet. So we didn't even start emptying. For solid meals, it might take 20 minutes to 30 minutes and the food is still, all of the food that you ate is still in the stomach. It didn't start emptying at all. But after it gets a bit grinded and mixed, it starts what? Going like into the duodenum. But for liquid meals, you can see, it's very smooth, no lag phase. Once you drink water, the water will start moving to the duodenum, like a little by little. Yeah. Like it's self-explanatory. I don't know why there's slides about this. Anyways, motility of GI organs. You can see it's either stomach, duodenum, large intestines. What I want you to know from this is that the stomach empties the fastest, and then the duodenum, and then the large intestines are the slowest to empty. Very easy. I think I wrote it somewhere here. Yeah. Stomach is the fastest, small intestines next, and large intestines are the slowest. So it goes in order. Yeah. Now for vomiting reflex. What's vomiting reflex? It's initiated by overdistension or irritation of the upper GIT organ. So if you ate something bad, or if you eat too much, and there is overdistension of the stomach, this vomiting reflex will be initiated. The afferent fibers that sense this distension are the autonomic nervous system fibers. They go to the center of the vomiting in the vomiting center that's found in the medulla oblongata. Okay. Now, this vomiting reflex will be initiated by efferent also autonomic nervous system fibers. So it's the same fibers that took it away, the sensory ones. They will bring it back by other fibers, but from the same nerves. Okay. Anyways, what's happening in this vomiting reflex is very self-explanatory. First of all, deep breath. Second of all, you elevate the larynx and close the epiglottis, and the upper esophageal sphincter relax. Do you remember the swallowing reflex? It's happening, but in like opposite. The main thing here, why do we close the epiglottis and all of that? You don't want the vomit to enter the larynx, صح? So you will close it. You will also seal the nasopharynx by the soft palate contraction. So no vomiting will go through your nose. So not through the nose, not through the larynx. What's the only pathway left? Exactly, through the mouth. What happens now? Diaphragm and abdominal muscles contract and squeeze the gastric content upwards. So they squeeze the stomach, and you know the gastric content will go upwards and go out of the mouth. This reflex can also be initiated by morphine. So even if there is no overdistension or anything, morphine can still stimulate this reflex, which is why, and it's not only morphine, like other chemical stuff can make you feel nausea and feel like you are gonna vomit because they stimulate these same receptors and same center. Questions? Okay. Take a pause if you wanna solve it. And yeah, it's E. Basically, it's asking when the parasympathetic nervous system stimulates, what happens to the slow waves? The spike potential frequency increases. If you remember, it was like this and one spike, but now it becomes like this, 300 spikes. So the spike frequency is increased. Please stop if you want to answer. And it's A. How does H. pylori affect directly? It's by ammonium. I know it increases gastrin secretion indirectly, but we are talking about the main thing is ammonium. Okay, so for the 7th lecture, it's a very easy lecture, inshallah, and you will understand it well, bidnillah. So let's take a look here. We have our pancreatic juice. First of all, let's agree that the pancreas is a gland. It has acini, it has ducts, like the salivary gland that we discussed. And the pancreatic juice secretion is going into the same 2 stages that salivary gland is going through. Acini releases and ducts modify. Easy? Easy. In the acini, we are still the same as the salivary glands. We are still producing this isotonic plasma-like blah 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 of juice that's full of ions and enzymes. Same thing. Now when it comes out to the ducts, here is the big difference that we will see. In the ducts, we secrete bicarbonate, like we did in the salivary glands. But this time, we are not secreting potassium. It's not here. It's only in the salivary glands. We are reabsorbing chloride, okay? Which is the same as we had in the salivary glands, salivary ducts, sorry. Yeah, it's part of the glands. Anyway, but we are not reabsorbing sodium like we did there. So potassium and sodium are out of the calculation here. We are only dealing with bicarbonate and chloride. This is the first main difference that I want you to have in mind. Second thing, in the salivary ducts, they were impermeable to water. So this caused what? A hypotonic saliva. Here, we are permeable to water. So when ions move, water moves with them. So what happens now? It's still isotonic, even when you are going through the whole thing. Even when you are modifying, it's still isotonic. Okay. Third thing that I want also to have, for you to have in mind, is that bicarbonate and chloride here are dependent on each other, which means that they move together. What do I mean by that? They have this transporter here, where we have chloride out and bicarbonate here. And we are getting bicarbonate secreting it and reabsorbing chloride. They are exchanging between each other. So what does that lead to? If bicarbonate is increasing and going out more, that means that chloride will go in more, so like reabsorbed more. And this we can see here, the effect of flow rate. Let's see what's the difference here between here and in the saliva. First of all, we said that sodium and potassium are not affected at all because they are not reabsorbed, neither secreted. So take them out of the calculation. We only have what? Bicarbonate and chloride. Bicarbonate increases with the flow rate. This means directly that chloride will decrease because you are reabsorbing it. If you remember, they are both dependent on each other. So if one of them increases the secretion, the other has to increase the reabsorption. So it's not dependent on the modification time that we have, like in the salivary ducts when we didn't have enough time to do modifications. Here, what increases the flow rate is a hormone called secretin. Remember it, guys? Yes, secretin itself. This is what increases the flow rate. When it increases the flow rate, it increases the bicarbonate. If you remember, the main effect was stimulating bicarbonate secretion. So chloride has to follow the same rule. So you will have this happening. So it doesn't have to do with the time that they have to do modifications and all of that we discussed in salivary glands. Now, for the osmolarity, as we said, it's not affected at all. It's still isotonic because water follows ions. You reabsorb, water goes. You secrete, water stays. So it's still isotonic. For the pH, of course, it will increase. Why will it increase? You are increasing bicarbonate, which is a base. So you increase pH. So yeah, this is a summary to what I just explained. You know, it has exocrine and endocrine, but we are talking about the exocrine part. Acini, pancreatic duct. Acini gives us this isotonic full of enzymes, fluid. In the ducts we modified. These are the main differences and all of that we have discussed. Now, digestion phases and pancreas. Do you remember the theory of digestion phases? Yeah, they will hunt us until like the end of the crash course. So, what happens to pancreatic secretions? They increase during all digestion phases. Very easy to remember. In the cephalic phase, vagus still simulates. In the gastric phase, nervous control also stimulates. And by the way, gastrin stimulates pancreatic secretions. So although secretin and CCK are anti-gastrin, gastrin is a good guy, so he still stimulates the pancreatic secretion. He doesn't care about what they do to him. Intestinal phase stimulates secretion via hormonal control, and we know this. The main stimuli in the intestinal phase, and this is the very important part here, acetylcholine, CCK, and secretin. CCK and secretin, you already know that and you remember them. Acetylcholine, we will talk about what does it do now. Let's compare these three guys. CCK alone increases mainly enzyme secretions in the acini. CCK, acini. Remember, CCK, acini. Now, secretin in the ducts. Main effect is on the ducts. So it stimulates bicarbonate and water secretion in the ducts. Does that make sense? So one increases the enzymes in the acini and one increases bicarbonate in the ducts. Acetylcholine is similar to CCK. When these three work together, they form what? Enzyme-rich solution. High flow rate because of secretin. bicarbonate concentration is elevated because of secretin. They form the perfect pancreatic juice. But if each of them works on their own, they will not do much, because you will have, for example, if secretin alone, you will have high bicarbonate solution, but it has low enzymes. CCK alone, it will has high enzymes, but it will have a very low flow rate. You get the idea. Secretin versus CCK, as we said, from secretin is from S cells, CCK is from I cells, if you remember. Secretin is mainly for the ducts, and CCK is mainly for the enzymes. What else? Okay. Enzymes are always formed in the acinar cell and stored in granules. Who stimulates their secretion? CCK, acetylcholine, as we discussed. Also other things that vagus does. So vagus, what does vagus give us? Acetylcholine, VIB, gastrin, they are all neurokines, right? Or neurotransmitter. Yes, all of these vagus stuff, they they enhance secretions of enzyme. Because you remember, parasympathetic is rest and digest. This last thing you don't need. Yeah, this is a very important part. Pancreas secretes all of the proteolytic enzymes, or all of its enzymes, in an inactive form. Yes. Why? Because if it's active inside the pancreas, it will eat the pancreas itself. So you want it to be inactive until it reaches out the duodenum. Now, we have our main activator. How are they activated? Okay, so they are now inactive. How do you activate them? The main activator is something called trypsinogen. Trypsinogen. It's an enzyme that's released from the pancreas in its inactive form, which is trypsinogen. This trypsinogen is then activated in the duodenum by this enteropeptidase, which is an enzyme of the brush border. Like the intestines themselves, they have enteropeptidase on it. So now enteropeptidase will activate trypsinogen and give us trypsin. Trypsin then proceeds to activate all other proteolytic enzymes. Easy, easy. So this was for proteolytic enzymes. What about lipolytic enzymes and amylase and all of that? They just need the alkaline medium to start work. Okay? Alkaline medium is enough for them to start working, so they don't need an activator. But the proteolytic enzymes use trypsin, which is activated by enteropeptidase. Now, as we said, trypsin secreted as trypsinogen and then activated by enteropeptidase. Why does it not affect the pancreas itself? Because we have protective measures that protect us from this. It is secreted in an active form. We have said that. Also, the pancreas contains pancreatic secretory trypsin inhibitor. This thing inhibits trypsin even if it's for any reason activated inside the pancreas. Also, the enzymes are stored in granules within the cells. So, they are like, you are closing them in a bucket or something. So, you are not afraid of them. Also, the duct's pH is higher than optimal pH for trypsin. Trypsin needs 7.5 to 8.5, and the duct's is always 8.5 to 9. As usually, so not always. So what happens if all of these measures are gone, and now it starts activating inside the pancreas? Acute pancreatitis. It's basically acute inflammation of the pancreas. Can be caused by gallbladder stones or heavy alcohol use, or self-activation of the enzymes inside the pancreas, which causes what? Autodigestion. That's what we call autodigestion of the pancreas, or self-digestion. You have a high level of serum amylase and lipase. Why? Because the cells of the pancreas have these enzymes. So when it starts getting digested and the cells burst open, the enzymes will flow through the bloodstream. So you'll have high amount of them. Fever due to inflammation, proteinuria due to inflammation, hypotension. These are things not very important. Now for Zollinger-Ellison syndrome, we have talked about it in P2, but I also want to again notify you about the secretin stimulation test. And that the point is that secretin usually decreases gastrin secretion, but here in gastrinoma, it for some weird reason, it increases it instead of decreasing. So you'll find that gastrin is increased. Also, if you give gastrin, synthetic gastrin, you will see no H plus increase. Why? Because it's already at the maximum. We have some questions here. Interpret test results. We have said that. Discuss the factor that can influence H plus secretion. We have said them, who does and who doesn't. Yeah, this one I want to keep in mind. Okay, we will just give two because these are the most two that we should know for now. And that is high acidity inhibits lipase, because if you remember lipase, which breaks down fats, needs an alkaline medium, right? So what will happen now? You will have inactive lipase because of the high acidity, because of gastrinoma, you have high acidity. And another reason is that high acidity destroys the mucosa of the duodenum, leading to malabsorption. And we will talk about that later. Don't bother with it too much for now. We'll see it inshallah later. Questions? Okay, so the answer is B. And basically, what activates proteolytic enzymes by pancreas? Of course, these guys are in the, what do we call it? In the stomach. So these guys are what we need. Just trypsin and enterokinase. So it will be B. Okay. The answer is A, cholecystokinin. Because, you know, fat, decrease gastric emptying. You know that cholecystokinin does that. Brush border, digestion and absorption. Bismillah. What is the brush border? Basically, you have these small intestines. Small intestines mucosa is folded, has these folds. These folds, we call it circular folds or plica circularis. These plica circularis have small villi. Can you see these things? Yes, they are, when you zoom in, you find them that are villi, that are made of these cells. Can you see it? Okay. Now these villi, when you zoom in, you will find that they have microvilli. Each cell of the villi has microvilli. You see this, subhanallah, this amazing creation of Allah gives us an increase in surface area by 600 times. The microvilli especially. And this makes, if you have 1 meter of intestines, it means like you have increased the surface area by 600 times. Can you imagine? And this gives it makes it the optimal place for absorption. These microvilli and all of this like orientation is what we call the brush border. The brush border is also very important because the enzymes that are the brush border digestion enzymes are usually found on the microvilli itself, so it's fixed. So we have pancreatic enzymes that are released from the pancreas, which are most of the enzymes that we use in digestion. And you have these enzymes fixed onto the microvilli, which are called enzymes of the brush borders. Anyway, digestion and absorption. From now on, we will talk about the 3 main things that we eat: carbohydrates, proteins, and lipids. And I will take you through each of them, how is it digested and how is it and how is it absorbed. But before we talk about how is it absorbed, we want to talk about the absorption sites. And this is a summary for them. Stomach takes the bad stuff, so ethanol, NSAIDs, aspirin. So it's not the major place of absorption for anything that we usually eat here. But most stuff is here in the duodenum and jejunum. Nutrients, vitamins, water, electrolytes, all of that is happening in duodenum and jejunum. Ileum is mainly for bile and vitamin B12. Remember this, please. Colon is water and electrolytes. Now, carbohydrates. We all said that we will start by how it's digested, then how is it absorbed. So, you take the starch, which is, for example, rice that you eat. It has starch. This starch gets broken down into maltose, which is a disaccharide. So, the first thing that you do is that you take this complex of polysaccharides, which is starch, and you break it down to disaccharide, which is what? Maltose. Or you can get directly sucrose, like the white sugar that we eat, or lactose from milk. Anyways, first thing you want to break down from big things and reach the disaccharide state. This stage is mediated by the enzyme amylase that we have discussed before. Okay? Okay. Once you reach the disaccharides stage, not place. Once you have these disaccharides, you will break them down even more. But how? By enzymes of the brush border. So this was pancreatic mostly, and from of course, the saliva. But here, it's mainly the brush border enzymes. What do? Take maltose, so it's maltase. Sucrose, sucrase. Lactose, lactase. Very easy to remember. And they give us these final products, which are monosaccharides. These monosaccharides can be absorbed. So, again, you will take the starch and break it down to maltose, which is a disaccharide, using these enzymes. Amylase, whether it was salivary or pancreatic. Mostly it's pancreatic. Maltose is a disaccharide. We have also other disaccharides, which are sucrose and lactose. If it's disaccharide, they can be broken down by corresponding enzymes of the brush border. Maltose and maltase give us to glucose. Lactose, lactase, glucose for galactose. Sucrose, sucrase, glucose for fructose. Easy. Now, we have these three final products that we came up with. Glucose, galactose, fructose, which are our monosaccharides. Now we can absorb them. We absorb them first into the cell from the apical surface. And then from the cell to the bloodstream, we send them down through transporters of the basolateral membrane. This is a summary to how we will do that. First of all, let's take a look at glucose. We have three things, remember, okay? Glucose, galactose, fructose. Glucose and galactose, similar in name and similar in mechanism. They use the same transporter, SGLT1. This SGLT1 takes one glucose in with one sodium. Please remember that. Same thing with galactose. So since they use the same transporter, they will compete for it. So if you have high amount of galactose, you will not be able to absorb glucose as much. And and vice versa. Fructose is a chill guy. He goes in very easily through GLUT5 without like competing with anyone. Once the three of them are inside the cell, they can go out from the other way through GLUT2 transporters. All of them can use it. And by the way, this beautiful thing, we will find it in every single cell. And its job here is to give us the gradient for Na. What do we mean by that for sodium? The sodium just got here with the glucose, right? If it accumulates here, the gradient will be high sodium inside the cell and it will go out. So we will not be able to absorb anymore. So this beautiful transporter kicks the sodium out to maintain the gradient. A summary for what I just said. Just two small notes that you will have that I didn't say now. Yeah. If this transporter SGLT1 is impaired, you will have osmotic diarrhea because you will not be able to absorb sugars. So it will accumulate here and it takes water with it. And if you take water out, it's diarrhea basically. Also, if the sodium potassium pump is inhibited, what will happen? As we said, Na will accumulate here and these pumps will be impaired. Na will not be able to come in. Very easy, okay? Now, lactose intolerance is basically when someone doesn't have lactase. Do you remember the lactase enzyme? If they do not have it, lactose will not be able to be broken down. So what will happen if lactose is accumulating in the intestines? Now bacteria will come and ferment it. You are not absorbing it. So bacteria say, okay, maybe I should do it myself. So they will come and ferment it. If they ferment it, they give us gas. And gases, basically, not one gas, okay? And this causes the bloating, diarrhea, blah, blah, blah. Why diarrhea? Because it's accumulating out and it's taking water with it. Now, proteins. Let's start with the digestion of the proteins. Digestion of the proteins happens on two phases, okay? Endopeptidases and exopeptidases. Endopeptidases takes the big proteins and breaks it down to smaller peptides. Can you see? An example of these are pepsin that we know in the stomach, trypsin, and like the big things that we know. Okay? Okay. Now when you have these small peptides, they are acted on by aminopeptidase and carboxypeptidase, which are exopeptidase. Okay? These exopeptidases, what do? They break down the small peptides into either amino acids or dipeptides or tripeptides or small intact peptides. We will talk about each of these. The main thing that we have endopeptidases that break down large proteins into smaller peptides, and then exopeptidases which break down these small peptides into even smaller stuff. Yeah. Now the absorption. What's happening here? We said that our final products are either amino acid, dipeptide, tripeptide, or small intact peptides. So how do we deal with any of these? If it's an amino acid, which is the small thing we could have, it goes in with an Na co-transporter, just like the glucose and galactose. Okay? So always the simple stuff, glucose, galactose, amino acids, will use this Na or sodium co-transporters. Okay? Okay. Now for di and tripeptides, they will go into the cell the same way, but instead of it being sodium, it's H+. Okay? Easy to remember? Easy to remember. Small peptides are the easiest ever. They are taken here by endocytosis and thrown out by exocytosis. No transporters, just endocytosis and exocytosis. Now, on the other way, now that we have them here in the cell, how do we get rid of them into the other way? Same way, but instead of it being co-transporter, it will be antiporter. So, do you remember we get amino acids inside using co-transporter with sodium? So amino acid and sodium, they both got into the cell. Now we throw it out and we get sodium in exchange of it. Same thing we do it here. It was co-transporter with H+ for di and tripeptides. It will be antiporter. Inshallah, it's easy to remember. Sorry, I think there's something I have missed somewhere here. Yes. When we are talking about here, the way of absorption, okay, it's very self-explanatory, but I want to note here that endocytosis mainly does not happen in adults, by the way, okay, when we just talked about. It's mostly in children, but in adults, it's mainly for vitamin B12. Other than that, it doesn't happen much, okay, even though we just did it happens in protein, but not much, okay. This is the thing. Now, yeah, this is just a summary. And these are the transporters to remember. Please also remember that the di and tripeptide co-transporter name is PEPT1. From the name peptide. Lipids. Lipids are the hard stuff. So please focus on them. Lipids are quite tough to absorb. Why? Because they are not as simple as carbohydrates or as proteins or anything else. So what do we do first? Lipids are hydrophobic. Lipolytic enzymes are found in aqueous solution, so they are mixed with water, which is hydrophilic, so they cannot mix well. So what do we need to do? We first do something that we call emulsification, which is basically turning big globules of lipids, can you see, into smaller globules. Why? Smaller droplets. Why do we do that? To increase surface area. First, the enzymes needed to work on the outer surface, only this amount. But now it can what? Work on this and this and this and this. So it's easier to absorb or to digest. Sorry. Okay, so emulsification, you what? Break down big molecules into small molecules using bile salt and lecithin. Bile salts and lecithin. So basically these are things that are found in bile, which is secreted. You know, we'll talk about it in its own lecture later. Now, okay, so we did emulsification. What now? Digestion is next. So, dietary lipids can be divided into three main types. After you emulsify it, it can be either triglycerides, cholesterol ester, or phospholipids. Triglyceride, cholesterol ester, phospholipids. These three, can you see them? They are dealt with with a certain enzyme for each of them, and they give us different end products. For triglycerides, they will be worked at by lipases. Very easy. The lipase that we know. Why I say lipases? Because we have lingual lipase, gastric lipase, pancreatic lipase. The main one is pancreatic, okay? And it turns it into monoglyceride and 2 fatty acids. Wasn't it 3? So now we have mono 1 and 2 fatty acids, 3 fatty acids. Cholesterol ester is worked by is getting digested by cholesterol ester hydrolase from the name, and it gives us cholesterol and fatty acid. Phospholipase from the name gives us lecithin and fatty acid. So all of them give us fatty acid plus stuff. So 4 possible end products, either free fatty acids or monoglyceride or cholesterol or lysolecithin. Now, just as a side note, we have an enzyme called colipase, which helps lipase attaching to these emulsified droplets. You will talk about it more in B10. It's B11, I think, Dr. Abdel-Jabbar. Anyway, so now we have our lipids, and they are now digested into these end products. What do we do now? Absorption. So, pancreatic lipase and colipase hydrolyze them into free fatty acids. Anyways, I want you to know that free fatty acids can be either short or long chain. Okay? So, short chain fatty acids will dissolve directly and be reabsorbed by simple diffusion through the membrane because they are hydrophobic, so they can go through the membrane easily. But if it's long chain fatty acid, monoglyceride, all other basically stuff in the products that we can't have, and also lipid soluble vitamins, they form these micelles before getting absorbed with bile salts, and they get transported to the brush border in order to be absorbed. Okay, micelles, what are their job? They are formed by bile acids surrounding, just like the emulsified droplets, wasn't it formed by bile acids? Now it's the same thing, but it's with the end products. It surrounds them and makes these micelles, which are basically small globules like this, which has our end products in it. And it's carried out to the brush border and it diffuses through the phospholipid membrane. Okay. Now, yeah, this is basically what we said. Here's the trick. After they are absorbed, there is another thing that we should do before getting them out of the cell, which is also another difference in fatty acids. Once they get absorbed into the cell, they can be bound to proteins, which is a big problem. Why? I'm talking about free fatty acids, by the way. If it happens, they will be more water soluble and they can damage the cell because they can dissolve. Free fatty acids are toxic to the cell. They should not dissolve in the cell under any circumstances. This is why after it's absorbed, okay, we reconstruct them. We take these free fatty acids, monoglycerides, and all of that, and we form the big stuff that we had at first again. After that, they form aggregates, and they are surrounded by what we call apoproteins. Do you remember outside when we formed the micelles and we had bile acids surrounding them? Now we have these aggregates, which are called chylomicrons, and they are surrounded by apoproteins. Okay? And now they can go out of the cell. As a summary, this is very easy now. We have digested and we got our end products. They are surrounded by bile acids to form micelle. This is what we call a micelle. They get inside the cell. We have now cholesterol, blah, blah, blah. Anyways, free fatty acids should not be in the cell. So what do? We reconstruct them into their original stuff. We form chylomicron, which is a big ball that has these guys inside of it, what we reconstructed, and it goes out through exocytosis through the lymph, and from the lymph to the blood. Now, once it gets to the circulation, how do we deal with it? Liver synthesizes low-density lipids. They take triglycerides to the cell. It's low, so they take them to the small stuff. High-density lipids, they get the excess cholesterol back to the liver because they are high, so they take it back to the big guy. Okay, you know the liver. So this is just a way to remember it. And we have lipoprotein lipase, which basically, while the chylomicron is going through the blood, hydrolyzes the triglycerides, and anyways, it digests the big molecules again into smaller molecules that can be used by the cells of interest. Okay. Malabsorption is when you can't absorb well. When does that happen? If you have problem with motility, so if the food is moving too fast, you will not have enough time to digest, absorb, do all of that. Or if there's digestion problems, which means that you don't have the enzyme responsible for this. This causes what? Malabsorption. Celiac disease. You might have heard of it. I think even some of you might have it because it's quite common, and it's basically sensitivity to gluten, and it's called gluten-induced enteropathy or something like that. Anyways, the problem is that gluten is a protein found in wheat. So when they eat bread or anything that contains this wheat, there is an autoimmune response from their body which destroys the brush border of the intestines, causing to malabsorption because they decrease the surface area, and it decreases also lipid absorption, causing staturia, which is lipid in feces, because you know there's decreased lipid absorption. So where will it go? Out of the body. Types of diarrhea. We have secretory diarrhea. We have mainly 4 types of diarrhea, and this is important, okay? I know them and know like the big examples that you will see. So we have secretory diarrhea. What is secretory diarrhea? When instead of the intestines absorbing water, they start releasing water. That of course will cause diarrhea. Easy to understand. What is an example for that? Cholera toxin is most important, and rotavirus. And I want you to keep this in mind. We'll talk about it in the next lecture, inshallah. And okay, for osmotic diarrhea is basically when like celiac disease and all of that and all of these things, or pancreatitis, where you don't absorb well, when there is malabsorption usually. If you do not absorb well, what will happen? This osmotic difference will take, for example, there is ions in the intestines, so it will drag water towards it, and if it drags water, what happens? Diarrhea. Motility related diarrhea. Also know the examples, and inflammatory diarrhea and the examples. Because why I'm saying the examples, I initially thought it's not important, but then she asked about this in the 17th batch exam. Okay, so apparently you should know them. I'm sorry, but it's mostly memorizing. What can you do? Now, what's the consequence of diarrhea? Why is it important to learn about it? We are losing ECF volume. We have drop in blood pressure, very self-explanatory. Metabolic acidosis, because you are losing bicarbonate. If you remember, feces are full of bicarbonate. Hypokalemia, because feces are full of potassium also. Clinical case and their answer. Easy questions. Okay, the answer is D, because it says sucrose, lactose, glucose, all of these guys can be absorbed well, no problem. But if it's starches that are ingested, they can't absorb it. Why? Because you will not have amylase. They have sucrase, lactase, everything that they need. But the problem is mainly with amylase, which is a pancreatic enzyme. You can't break the starch. Now this. Yeah, it's secretory diarrhea. As we said, VIB is related to secretory diarrhea. Okay. So, water and electrolytes absorption. This is very easy and quick lecture, inshallah. So let's start with jejunum and what's going on in each of them. Starting with jejunum. So first of all, I like to start from here. Our beautiful reaction that gives us hydrogen and bicarbonate. Hydrogen will go out of the cell through this exchanger. So what did we just do? We reabsorbed what? Sodium. And also sodium can go in through with sugar or amino acids like we discussed before, either from the SGLT1 with sugars or with amino acids like we discussed. So we reabsorbing sodium. Beautiful. What happens to the bicarbonate? Goes directly to the blood. So we reabsorbed what? Sodium and bicarbonate. Easy? Very easy. The difference is here. Look at the ileum. Same thing is happening. We have our H and bicarbonate. H goes out in exchange of sodium. Sodium goes in through multiple ways and it's reabsorbed. But bicarbonate does not go out to the blood this time in the ileum. It gets exchanged for chloride. And chloride is the one getting reabsorbed. So what did we do? We reabsorbed in the jejunum sodium and bicarbonate. In the ileum, we reabsorbed chloride instead of bicarbonate. Easy? Easy. Now for colon. Colon is the same as late distal tubule. You have sodium absorption, K plus secretion, and it's so similar that aldosterone affects it. What does aldosterone do? Increases sodium absorption, increases K plus secretion. Very easy. Colon electrolytes secretion. The colon does not only absorb, it also secretes. And this could be a problem. Why? Let's look here. We have this beautiful transporter. Can you see? This loads the cell with Cl. Because each time it works, you have 2 Cls going in. 2 Cl. So we have high amount of Cl here. Does that make sense? Okay. What's the problem then? These Cl ions will go out of the cell through this CFTR channel. Okay, beautiful. And this is normal. The abnormal thing here is when you have cholera toxin. Look at what cholera does. It goes and it activates an enzyme called adenyl cyclase. Okay, I will not go through the G protein pathway. We are not a mod. Okay, just know that adenyl cyclase is activated. Adenyl cyclase gives us more cyclic A and B. You know that. What's the problem is that cyclic A and B activates this CFTR channel. Normally, it doesn't work much, CFTR channel, because you don't want to secrete much colon. But cyclic A and B keeps it activated. Cholera toxin increases this activation, causing an overactive CFTR channel. So what happens here? Chloride will be secreted abnormally, like a lot of chloride will be secreted. Who follows ions? Can you remember? Yes, it's water will follow. And by the way, in the colon we have tight junctions because she asked about somehow. So these are tight junctions, but water can also follow. Easy. Now another thing is that VIB can also increase adenyl cyclase and give similar effects. This is why, if you remember when we talked about secretory diarrhea like 2 minutes ago or something, we said that VIB can also affect. Does that make sense now? Okay. This is a summary for what's absorbed, what's secreted. I cannot say much. Most of it you already know. What you don't know yet is maybe that calcium and iron are absorbed in the duodenum and that. I just want you to give you this mnemonic. You can remember. Dude is just feeling ill, bro. This is for iron, folate, and B12. To remember where each of them is being absorbed. So dude is duodenum, iron, just feeling, jejunum, folate, ill, bro, ileum, B12. Just to remember that, in ileum, B12, and duodenum, iron. Folate is not here, but just, I thought it will be helpful. And yeah. Now, fluid retrieval from GIT in hypovolemia. When we have hypovolemia, we have renin-angiotensin-aldosterone system activated, and sympathetic nervous system, fight or flight. When you have hypovolemia, of course, you want to activate that. So what happens? It stimulates reabsorption through vasoconstriction. You don't need to know the mechanism, just know vasoconstriction by sympathetic nervous system simulates reabsorption. Renin-angiotensin-aldosterone cascade helps how angiotensin stimulates sympathetic nervous system. So this goes to this again. And it gives us also aldosterone, so it gives us this. What does aldosterone do? Reabsorbs sodium, and who follows sodium? Water. And that also, of course, happens in the colon, as we just discussed before. Absorption of minerals. Minerals are absorbed without digestion, like just right away. Because where would you go? For example, the mineral is iron. Will you break it into electrons and protons? Of course not. So yeah, it's not digested. The thing that you should keep in mind is that iron and calcium are different than other minerals. Why? Because they are absorbed in proportion to the body's need. So if you need more, it will take more. If you need less, it will take less. Other minerals have constant rates. So they do not care much about whether you need or you don't need. It's not their concern. Now, iron absorption, very easy. I just want you to know that it goes into the cell through DMT1 channel and goes out through ferroportin. And I want you to know that whether it's coming in or going out, it has to be in its first form, Fe2. If it's in Fe3, you need to reduce it first to Fe2 and then absorb it. So if a question asks, Fe2 or Fe3 are easier to absorb. Of course, it's Fe2 because you can absorb it right away. Fe3 needs to go through a long process before it's reabsorbed. Calcium, self-explanatory, to be honest. Calcium channels, it goes out through many pathways, either with exchange for sodium or what do you call it? By ATPase, yani. The thing that you should also keep in mind is that calcium is regulated by vitamin D3. You know that calcium, vitamin D3, they go along beautifully. But the thing is that vitamin D3 affects only transcellular pathways, so only the thing that's happening here. Calcium can be reabsorbed paracellularly, and of course, vitamin D3 does not have an effect on that. This is summary for sodium, potassium, chloride, water absorption. They go through 3,000 channels that we have discussed in each place or whatever, and they go the other way through the ATPase pump. And we have water and potassium moving paracellularly. Okay, nothing much. This is a summary that you can take a look at if you want. I think we talked most likely about all of that. We have this clinical case. And with the answer, I just want to note that here, what location the gastrointestinal tract has tight or impermeable junctions between the epithelial cells. It's large intestines and also stomach, but the doctor didn't mention stomach. So, that's weird, but it both works, to be honest. For this question, just say large intestines because it was asking about things about diarrhea and all of that. Now, which of the following is the rank order from gas to least of areas of water absorption in the GI tract in a normal individual? Yeah, it's small intestine, colon, then stomach. I think I mentioned it in the slides. Maybe I forgot to say it, but basically small intestine. We will discuss it in Dr. Abdelrahman's lecture, inshallah. So don't worry about it. But for now, just know, small intestines, colon, stomach, they go this in this order. Now, the answer is E. It's saying cholera-toxin affects which step directly. Of course, it will be the CAMP step because it increases CAMP concentration by activating adenyl cyclase. Don't say B. Yes, B, the chloride secretion will increase, but that's not the direct effect. The direct effect is on CAMP, cyclic AMP. Okay? Did I put it twice? Yeah, I'm sure. Now for the 10th lecture, which is the start of Dr. Abdel-Jabbar lectures, we'll be talking about biliary secretion. I just want to say thank you, Malik, before we start. He's the one who did the slides for 10, 11, and 12. So thank you so much, boy. I couldn't have made it without you, so thank you. We will be starting by lipid digestion, okay? It's not a part of the lecture, but I just want to refresh your memory about how lipid is digested and the role of bile. We have these big fat globules, and by emulsification with bile salts and lecithin, which are components of bile, they become these emulsified small droplets, okay? Now these small droplets will then be digested using lipases and colipase. After that, they will become these end products, which we have talking about. Fatty acids, for example, and monoglyceride and all of that. They are then forming mussels. Also, bile salts are what help in forming these mussels and bringing the end products into the membrane. As you can see, an overview. These are our mussels with our end products surrounded by bile salts. They are brought into the membrane, then they reconstruct again, if you remember, in the small endoplasmic reticulum, and then they form these chylomicrons with the apoprotein, and they are transferred out of the cell to the lymph. This is all you should already know from Dr. Angelica's lecture, which is here, P9, I think, or P8. Anyways, now for the actual lecture. What is bile? Bile is secreted by hepatocytes, modified in the ducts by cholangiocytes, stored and concentrated in the gallbladder and released into the duodenum. I like to think of it like the pancreas, for example, or the salivary glands. Where, the saliva, for example, was secreted initially from an acini. Our acini here are the hepatocytes, just as a way to think of it. It's not very accurate, but yeah. And then it gets modified in the ducts, right? Same thing here. Cells of the bile ducts, which are cholangiocytes, are modified. The quite difference here is that we store it and concentrate it in the gallbladder. Okay, so this is the role of the gallbladder, concentration of bile. And then it gets released into the duodenum. How? You contract the gallbladder and relax the sphincter of Oddi. And yeah, then once it reaches the duodenum, you know what it does. We just said the lipid digestion stuff. Now, I want you to know that not all of bile will be newly made, okay? Most of the bile that's secreted into the lumen, or into the duodenum, will then be reabsorbed in the ileum and in the colon. Like 95% of that will be reabsorbed, and it goes back into the liver, like recycling. So the liver only synthesizes the lost amount of bile that will be excreted to feces, and we will talk more about that later. This is just an overview. The main component of bile, okay, is bile acids. Bile acids. Where do we get these bile acids from? Cholesterol. We get the cholesterol, and by an enzyme called 7-alpha hydroxylase, we will be talking more about it later. They give us two, what we call primary bile acids. It can be one of two, either cholic acid or chenodeoxycholic acid. I know it's a long name, but okay, you have to remember it. Cholic acid, chenodeoxycholic acid. These are our primary bile acids. Then it gets conjugated. Conjugation basically means that you are adding something, whether it was amino acid or glucuronic acid or a sugar, anything that you add just to make it more water soluble. And why am I giving this general thing? Because in bile we will conjugate it by something, in bilirubin we conjugate it by other thing, but the main purpose is one, increasing water solubility, and I will discuss it in more details later. Now, after it gets conjugated and it's becoming more water soluble, it can go and be stored in the gallbladder, or it can be, and then it will be released into the duodenum, or it can be released into the duodenum right away. The thing is, after it's in the duodenum, you know what it does, its job, and at the end of the day, some of this bile will be deconjugated again. Okay, and we will be talking about that, and then all of 95% of the bile, whether it was deconjugated or not, will be reabsorbed into the hepatic circulation. This was just an overview. I don't expect you to understand everything here for now. Now, what does bile contain? As you can see, the main thing is our bile salts, which are our bile acids or whatever. You can see that it also has some cholesterol. Even though bile salts are made of cholesterol, it doesn't mean that all cholesterol will be taken to bile salts. We will have a bit of cholesterol there, and we will discuss why is cholesterol important here and what's the clinical correlation for that. Okay. You can see also that it is isotonic to the plasma, so the amount of ions are almost the same. You can see just that it has higher bicarbonate concentration than the plasma. Okay, so bicarbonate here is more, and that makes the pH alkaline. Okay. Now let's go back from our beautiful cholesterol and recap what we said a few minutes ago. We have our cholesterol. What happens is that we have this enzyme, 7-alpha hydroxylase, rate limiting step, by the way, or rate limiting enzyme. It gets this cholesterol and it adds hydroxyl group from the name hydroxylase, or it adds hydroxyl group and a carboxyl group. Why? What's the point? To make it more water soluble. Now we have formed our primary bile acids, can be cholic acid or chenodeoxycholic acid. Easy. Now, what happens after is the conjugation, as we have said. we are adding something to make it more water soluble. This is what we said, right? So what we will add, glycine and taurine, which are amino acids. I know taurine is not very known amino acids, not one of the 20 that you know, but it's still. We are adding glycine and taurine to conjugate the bile acids, and they are still called primary bile acids, but they are conjugated. Once they are conjugated, you see the name is easy, and you are adding taurine to chenodeoxycholic acid, so it becomes taurochenodeoxycholic acid. You are adding glycine, it becomes glycocholic acid, acetylic acid, it's easy. And as we said, it becomes more water soluble. How? It's something that has to do with the pKa and the bile acids being more ionized, and more ionized means water soluble. But I don't want you to dig deep into that. It's not that high yield of a point, okay? But just understand that conjugation changes the pKa, makes it more ionized, so more water soluble. Okay? And always remember, conjugation, water soluble. Now, deconjugation. I want to note that this deconjugation that we will be discussing only happens to 5% of the bile acid that we have. Only 5% will be deconjugated. Most bile acid will be reabsorbed as conjugated bile acid, and we will discuss it now. So what is deconjugation? Basically, we have in the ileum these bacteria. These bacteria can deconjugate the bile acid. From the name, what will it do? First thing, it will take off the amino acid that we added. And that makes sense. But the bacteria doesn't stop at that. It even takes out the hydroxyl groups. So it's as if it came back to cholesterol before we did anything. And that's not correct, by the way. I'm just putting it into perspective. So just to understand, the bacteria made it so hydrophobic. It took out everything that made it water soluble. So it became what? Hydrophobic. Why? To make it more absorbable for the membrane. And we will discuss it in a moment. So as we said, the bacteria deconjugated. Okay, just something that I forgot. Once it's deconjugated, we call it secondary bile acids. Secondary bile acids when it's deconjugated. Does that make sense? So primary, after we just made them, they are primary. Once we deconjugate them, they are secondary bile acids. Now, as we said, less water soluble, more lipid soluble, more absorbable. We will be talking about that. And I want you to know that 90% of the bile acid will be reabsorbed in ileum, and 5% in the colon, and 5% will be excreted. We have said that. And this is something that you should keep in mind. The secondary bile acids that are coming after the deconjugation are two things. Please memorize the name also. Deoxycholic acid and lithocholic acid. This is the more important one. Why? Because a question came about it, and it's about this information. Lithocholic acid is the most found in feces. It's not absorbed well, okay? The absorption of lithocholic acid is very poor. So it's mostly the one being found in feces. Lithocholic acid. Now we have our beautiful muscles. You know the muscles. I don't know why they are here now, but but just to understand the job of bile salts in forming muscles, bile salts, lecithin, nothing much to know. You already know the muscles. This is the hepatic biliary system. You don't need to know the details here. I just want to show you something, and it's here, yeah. When you are looking at the hepatocyte, you will see this. This hepatocyte has inside of it what we call bile canaliculus. Yes, canaliculus is one, and more than one of them is called canaliculi. Anyway, so we have these bile canaliculi, or canaliculus, or whatever. These bile canaliculi, they join together to form bigger bile ducts or bile ductules, and then bile ducts and all of that. So they are doing the merge that Dr. Annie talked about. Why are we talking about this? Because I want you to know that each hepatocyte is releasing the bile that it's making into this bile canaliculus, and then it joins together to give us bigger and bigger until we reach the bile duct. Does that make sense? Okay, easy. Now we have discussed that we are recycling the bile that we are making. So in the ileum, it will be reabsorbed mostly, right? And we said it can be either deconjugated by the bacteria or it can be not conjugated, which is most of the bile. So what now? It's going back to the liver, right? So it has to have a transporter to enter the liver, the hepatocyte, I mean. These transporters are as follow. The first transporter you will find is NTCP transporter, which is for conjugated bile salts to get into the hepatocytes. Okay? Which is most of the bile salts that will be recycled. They come by this transporter, which is what coming with a co-transporter with the sodium. And you can remember it from the N. This N is for Na, sodium. Now the other transporter will be for unconjugated bile salts that are coming into the hepatocytes. Can you see that? Okay, this transporter is the one used for unconjugated bile acid. Now, if it's conjugated, it can go out of the liver right away because it's already conjugated. If it's not conjugated, you will conjugate it first. But the point is, after it's conjugated, you will secrete it again into the bile canaliculus. How? By this transporter. And this is the rate-limiting transporter that's found in the hepatocytes, in this hepatic circulation thing. Basically, it controls how much is secreted, so it's the rate-limiting one. And it uses ATP. The thing is, it takes this conjugated bile acid and throw it into the bile canaliculus. So, makes sense? This is our 3 transporters that are found here that I want you to remember. These guys are not important. Okay? I hope that makes sense. Now, just a side note for people who speak Arabic. This you can remember by the name is basib. Basib is I leave in Arabic. And basically, it's leaving the hepatocyte. If you do not speak Arabic, just memorize it. This is not for you. Anyways, the modifications that are happening in the bile duct. We said that we will secrete it from the hepatocytes, and we understood what happened. Now, what happens to the bile that's secreted in the bile ducts? We will modify it, right? First of all, this is a cholangiocyte. I want you to see something. We want to have our result, the bile that we want to secrete, needs to be dilute and alkaline. Dilute and alkaline. How will we do that? Look with me. First of all, we will load the cell with chloride by this transporter. Please don't mind the numbers, just focus on me. So we will load the cell with chloride by this transporter. So we have a lot of chloride here. We will also load the cell with bicarbonate by this transporter. Okay, so we will have a lot of bicarbonate here. Now what? This chloride will leave throughout this channel and go out, which is the CFTR channel, and fill this side, which is the lumen, with chloride. And then it gets reabsorbed through this transporter in exchange of what? Bicarbonate. So what did we do? We threw out bicarbonate outside. Can you see that? Because you want to make it more alkaline. Who does that job? Of course, bicarbonate. So we will load the cell with chloride and bicarbonate, and then you exchange chloride for bicarbonate. But how will we get the chloride out? Through the CFTR channels. Does that make sense? Now, since you are throwing ions in this side, chloride and bicarbonate, all of that, water will follow. Whether it was paracellularly or through aquaporins, for the osmotic defense. And now we have water and bicarbonate on the other side, making the bile what? Dilute and alkaline. Make sense? I hope that makes sense. Now for the gallbladder. Once the liver, what's this? Yeah. Once the liver secretes bile, it can go to 2 pathways. 70% of the bile will be going to the gallbladder itself. Okay? But as you can see, it's not like a closed road that the bile needs to follow. It can also spurt this way. And the pressure is high to the point where there can be a spurt of bile into the duodenum. So a portion of the bile will go into the duodenum. And that's most likely correlated to motilin. Okay, motilin is what makes this effect, but you do not need to remember that at all. not at all, but it's not a very important point. Anyways, you just need to know that 70% will go to the gallbladder and 30% will spurt into the duodenum because of the high pressure, even if the sphincter is closed. Now, what's happening in the gallbladder? We said we will concentrate the bile, if you remember. So what does it do? To concentrate it, it takes out water. It takes out water. So can you see the bile salts here? It's 1 in the liver. In the gallbladder, it's 6 because we took out a lot of water. It doesn't add bile salts. No, it just takes out water. So the concentration increases. But you will see that everything happens like this except for these 3 ions. Okay? Can you see? Chloride, sodium, chloride, bicarbonate. So we reabsorb. Why? Because they are also reabsorbed with the water. So we reabsorbed what? Salt, water, and bicarbonate. Does that make sense? And we will talk about the pH in a minute. But you can also interpret that it will be more acidic because we lost bicarbonate, which is our base. Now, how is this happening? Sorry, guys. You can see this is not my first record. Okay. What's happening in here? I will tell you now. We want to reabsorb, if you remember, sodium chloride. So how will that happen? From the lumen, we have our transporters. Okay, 1 for sodium hydrogen and 1 for chloride bicarbonate. And this transporter will cause the reabsorption of sodium and chloride. An exchange of what? Hydrogen and bicarbonate. Okay, what about water? What's happening? Well, when you secrete these two, hydrogen and bicarbonate, do you remember what they do together? Yes, they form water and carbon dioxide. Carbon dioxide no one cares about. But the thing is, water will be reabsorbed through these aquaporins. So you also reabsorb water. And it will also follow the osmotic gradient because, you know, the sodium and chloride will be thrown this way. Now, something that I want to note here is that the acidity of bile happens because of this. Now we have hydrogen here and bicarbonate, and they neutralize each other, so the pH should not be any different. But the thing is, we have less of these and more of these transporters. So that causes this. Let's say we have 10 hydrogen ions, and we have 5 bicarbonate. 5 from the bicarbonate will go with 5 from the hydrogen, leaving us with 5 more hydrogen. Okay? These 5 will form us water and all of that, and it will do nothing. But we'll have 5 more hydrogen staying here. And if you have hydrogen, what do you have? Lower pH, which causes the acidification of bile. So if a question comes, what causes the acidification of bile in the gallbladder? You know, more hydrogen sodium exchangers than chloride bicarbonate exchangers. Easy? Easy. Now, this is self-explanatory. I mean, you know, after the meal, the sphincter will open and the bile will contract to throw the bile out. Now, this is the enterohepatic circulation that we were talking about more than once, and lithocholic acid that you will find in feces and all of that. And as you can see, 600 milligrams will be synthesized daily. Why? Because these are the same 600 that we are losing in fecal execution. The rest of bile is what's going on with it? It's reabsorbed. Right? Now, this is the enterohepatic circulation that we have talked about, and we said that the bacteria deconjugate some bile and some of it will not be conjugated. So what does that lead to? The bile that was deconjugated is now very lipophilic and very hydrophobic, right? So it can diffuse through the membrane and it will be reabsorbed in the intestines this way. But what about conjugated bile? How will it be absorbed in the intestines? By these transporters. Can you see? ASBT gets our bile acids with sodium into the cell, the intestine, and then bile acid will go out through OST channels. I just want you to remember the names. And then they go to the portal circulation and blah, blah, blah. Now, regulation of bile synthesis and secretion. It's very... it makes a lot of sense what happens. Subhanallah, we have this mechanism where we regulate everything, you know that. So, if we have high levels of bile acids, okay, just a minute, yeah. High levels of bile acids in the hepatocytes. you remember how did we make bile acids aslan? By this enzyme, right? That took cholesterol and turned it into primary bile acids. Yes. So what will happen to this enzyme? It will be inhibited because we already have a lot amount of bile acids, right? A high amount of bile acids. So you don't want to make any more. It will be inhibited. What if you have low levels of bile acids? It will be stimulated. Makes sense. طيب, what about this channel? Do you remember this channel? Nasib, leaving the cell? So it's the one that's throwing bile into the bile caniculi from the hepatocyte. It will be stimulated here because we have high amounts. So we don't want the hepatocytes to be overstimulated by this bile. We want to throw it away. So it will be stimulated. But if we have low levels, no, we don't want to lose this bile, so we will keep it. So it will be inhibited. Easy, very easy. Effects of cholecystokinin. You know the effects of cholecystokinin, which is the contraction of the gallbladder and the relaxation of sphincter of Oddi. But how is it doing that? Well, to your surprise, it's a hormone, but its endocrine effect is actually minor. The main mechanism where it affects is neural effect. And please, there was a question about this. I forgot to put a star here. But anyways, there was a question about this. And it's the main effect of cholecystokinin is being done by vagus-vagal reflexes. Cholecystokinin stimulates neurons. And these neurons will then, by vagus-vagal reflexes, afferent, the reflex. It will do a reflex and cause this contraction of the gallbladder and relaxation of sphincter of Oddi. Does that make sense? So yeah, cholecystokinin, main mechanism is vagus-vagal reflex. Gallbladder function, this is just a summary for everything. We have talked about this more than one lecture, so this is a summary. And now gallstones. Do you remember when I said there was a bit of cholesterol in bile acids? Sorry, not bile acids, in bile itself. Now, here is the problem. Gallstones. What forms gallstones? Gallstones are formed mainly by cholesterol. So they are what? Cholesterol. So how do they form? If cholesterol precipitates, it's formed. But how do we prevent that? We have two players that are helping us to prevent that. Phospholipids and bile acids. So the thing is, cholesterol is trying to precipitate, bile acids and phospholipids are telling it, no bro, stop. So if these two are decreased, the phospholipids or the bile acids, or the cholesterol is increased, it will cause gallstone. And we have a ratio that we use. Can you see that? 10, 3, 1. What does that mean? For each one cholesterol, you need to have 3 phospholipids and 10 bile acids. This ratio, if it's kept maintained, then no gallstones will be formed, inshallah. But what if cholesterol becomes more? It becomes 2 to 3 to 10. This is dangerous, because then it might precipitate. What if the cholesterol is the same, it's 1, but phospholipids are decreasing? Still dangerous. What if the phospholipids and cholesterol are the same and bile salts are decreasing? Still dangerous. Okay, makes sense? Very easy. And there was like 7 questions about this in the past. So please keep it in mind. Now gallstones. Gallstones can block 3 sites. Either, they are made in the gallbladder, gall from the name. So they either block the cystic duct, which is the duct of the gallbladder. What will that lead to? Bile is still coming from the liver. It will try to go to the gallbladder, not working. It will continue and it's okay. So does that affect bile salts delivery to the duodenum? No. Bile salts will still go to the duodenum, but without moving by the gallbladder. Does that make sense? What about pancreatic enzymes? Still, the pancreatic duct is very far away from the blockage. Make sense? Makes sense. So what are the complications? Just the problem with the cholecystitis. This is what we call it. Now, what if it blocks the common bile duct? Here. Now comes the problem. Because the liver will try to send the bile. Some of it will go to the gallbladder and all of that. Anyways, when you want to excrete that bile into the duodenum, it will find the blockage. It cannot bypass it. So what will happen? The delivery of bile salts will be decreased, and this causes jaundice. And steatorrhea because you will not reabsorb. You will not, digest lipids. Okay. What if the blockage is happening at the sphincter of Oddi or the ampulla of Vater or whatever? Now, before that, sorry. When the blockage is here, the pancreatic enzymes can still deliver can be delivered to the duodenum because still the pancreatic duct is far away from the blockage. Now, what if the blockage is in the ampulla of Vater or at the sphincter of Oddi? Here is the biggest of problems because now the bile is blocked and so is the pancreatic enzymes. And that could cause pancreatitis, jaundice, cholecystitis, all complications that you might think of. Does that make sense? And yeah. Okay, so for the 11th lecture, we will be talking about the liver metabolism and bilirubin. Again, thank you, Majid, because he did the slides also. I want to start from here, which is a slide I took from Dr. Khalid for histology, because I want you to understand something. This is a liver lobule, which is made of collection of liver cells that are oriented in some way. I don't want you to know from here except one thing. Just look here. You can see that we have this side of the lobule and this side of the lobule. This first side is closer to the what we call the portal triad, right? Where we have the hepatic artery and portal vein and bile duct. Okay, branches from them, of course. Now, this side, on the other hand, is very close to the central vein of the lobule, which is a normal vein that's like the hepatic vein. Why is this important? Here. You see, here is our first part that we were talking about, and here's the second part that's close to the central vein. The first part, since it's close to the hepatic artery, it's receiving oxygenated blood. This part is very far away, and it's actually closer to the central vein, so it gets low oxygen. The first part we call zone 1, which is closer to the portal triad. The second part we call zone 3, which is closer to the central vein. So what's happening in each of these zones? The cells that are close to the oxygen do oxidative phosphorylation because it needs oxygen, and they have it. They also have their uptake of bile acids because the conjugation, all of that, oxygen is needed. And they also have Kupffer cells, which are macrophages that we'll be talking about of the liver. On the other hand, the cells that are here, that are receiving low oxygen in zone 3, are doing anaerobic glycolysis because they don't have oxygen. They do the detoxification, okay. And they are sensitive to ischemia. This is a very important part because if they can't afford to lose any more oxygen. They already have very low levels of oxygen. So the first part of the liver that's damaged when there is low oxygen than usual is zone 3 because it already has low oxygen. So any less than that and it's gone. Kupffer cells. Kupffer cells are macrophages of the liver, basically. And they are sitting in the sinusoids of the liver and they phagocytose anything that foreign material that might get into that sinusoid. And this is summary for the cells of the liver. You see we have our beautiful hepatocytes. They have the bile canaliculi. They do the metabolism and they regenerate. And regeneration is a very important thing because, when someone donates their liver and it grows back, we all heard about this before, right? This is because of this ability of regeneration that they have. Also, stellate cells are very important, and there has been a question about their vitamin A storage capability, okay? So know that they are the one that store vitamin A, and they produce collagen after injury. So they are cells responsible for fibrosis. Have you ever heard about liver fibrosis? Yes, stellate cells are the one responsible because they make fibers, collagen fibers, after injury. Now, for Kupffer cells, as we said, they are the phagocytes of the liver. We have our endothelium cells, which are sinusoidal, so they are very permeable. And we have our cholangiocytes that we have talked about before, which are cells that form the bile duct, basically. Now, for these functions of the liver, you don't need to memorize them or anything. Just know that it participates in metabolism of a lot of things, and we will talk about a few things now. Starting with bilirubin. What's bilirubin? I will love to explain it here. It's better. Look, we start with our red blood cells. Our red blood cells get old and we want to get rid of them. Okay, how do we do that? We break them down, we get hemoglobin. Hemoglobin, we take from it the heme part especially, okay? And this is happening in the macrophages, of course. They are the one getting rid of these red blood cells. Heme is turned into what we call unconjugated bilirubin or indirect bilirubin. Please remember this. And it's water insoluble. So what's the problem? Since it's not water soluble, we can't excrete it, sorry. So what's the solution that, subhanAllah, we have? Is that we take this unconjugated bilirubin and we turn it into conjugated bilirubin. How? It's attached to an albumin protein in the blood that we have heard about before, and it goes to the liver. The liver does the conjugation like it did to the bile acids, yes, the same way. But it uses this time what we call glucuronic acid. It's not an amino acid. But do you remember what I said in the last lecture? Conjugation means water soluble. So the point is that we want to make it water soluble. What's the enzyme that conjugates it? Please remember it. There was a question about it before. UDP, glucuronyl transferase. Okay? Or UGT for short. Now, this conjugated bilirubin is now water soluble. So it will be, it can be secreted in the bile. Then, once it reaches the intestines, the same thing that happened with the conjugated bile acids happened with the conjugated bilirubin, which is the deconjugation by gut bacteria. This turns it into urobilinogen. Urobilinogen is a clear substance, like it doesn't have a color, but then it has more than one fate. It can either end in the feces as stercobilin. Stercobilin is brown in color, and this gives the stool its color, the brown color, and you can correlate it by stercobilin stool. Please remember that. This is most of urobilinogen. What's left from that, a portion of it will be excreted by the kidney as urobilin, which is yellow in color, and this is what gives the urine its color. And you can also correlate it by urobilin, so it's urine. I hope that makes sense. The bigger portion of what was left originally, this 20%, the bigger portion of it will go into the liver, into hepatic circulation, and be conjugated again and excreted, and so on. We will try to get rid of it in the next cycle. I just want to note that this diagram is missing something, which is that heme does not turn into unconjugated bilirubin directly. As you can see here, it becomes first biliverdin. It's not very important, but just to know the pathway well. Biliverdin is a green-looking substance, and then biliverdin is turned into bilirubin, or unconjugated bilirubin, as we said, and it's a yellow-looking substance. And I want you to emphasize this yellow color. Why? Because we are talking about bilirubin because of jaundice. Jaundice is when the person is basically yellow. Can you see this baby? He's looking yellow, right? Look at the eye of this lady. Yellow. This is jaundice. And it's caused by bilirubin. Why? Because it's yellow. If we don't excrete it, it will accumulate and cause this. Okay, we said we have two types of bilirubin. So which one does the problem? Both. But we differentiate of the causes, we differentiate the causes of hyperbilirubinemia, which is a lot of bilirubin, based on if it's unconjugated or conjugated. If it's unconjugated, we call it prehepatic. Why? Because where is the unconjugated bilirubin? Is it before the liver or after the liver? Like in this pathway, when you look at it, just to clear it up for you, you will see it's before the liver. Once it reaches the liver, it gets conjugated. But here we have our unconjugated bilirubin. So the problem will be before the liver if what's high is unconjugated, such as hemolytic anemias with all their kinds, sickle cell, G6PD deficiency, all of these. Any hemolytic anemia that causes a lot of heme to be, red blood cells to be broken down, and hence more heme, and hence more unconjugated bilirubin. Okay? Our second type is posthepatic, and it's conjugated bilirubin. We call it obstructive jaundice. Why? Since it's conjugated, it means that it had reached the liver. But the problem is after. There is a blockage here that forbids the conjugated bilirubin from leaving the body. Does that make sense? So it will be a problem post-hepatic, after the liver, such as gallstones. It means the bile isn't moving out because of gallstones, biliary tract disease, any problem with the excretion of bile. Okay. Now for urobilinogen in urine, if it's more than normal, it will be hemolytic anemia, and it makes sense because you are making more of this, until you have more urobilinogen than usual. Okay. Now what about if it's less than normal? It will be obstructive jaundice, post-hepatic. Why is that? Because you will have blockage here, right? We said that. So the conjugate will accumulate and it's not turned into urobilinogen, so it will be less. Does that make sense? I hope it does. Now, NSAIDs. When someone takes NSAIDs, which is non-steroidal anti-inflammatory drugs, whatever, they are taking basically this substance mainly, okay, which we will take for short, APAP. This APAP will get conjugated, 90% of it will be conjugated and turned into non-toxic substance. I don't want you to remember what exactly it's turned into, but just know it's non-toxic, which is good. 2% will be excreted as urine, so also no problem. The main problem lies here. Can you see? 5 to 9% of that will be turned into what we call an NAPQI. What I want you to remember from this is that this NAPQI causes oxygen damage due to free radicals. Free radicals means oxygen damage. If you remember more, yani. Anyways, what neutralizes these free radicals? GSH, which is reduced glutathione. If we have lots of it, we will be able to neutralize this effect, and inshallah there will be no damage. But if we don't have lots of it, then we will form a lot of free radicals, ending with liver damage and possibly liver failure, which, as you can see, up to 50% of overdose-related liver failure is due to that. Not alcohol, but it's NSAIDs. Can you imagine that? Oh, of course, alcohol too is a big no. Okay. By the way, which zone is affected? It's zone 3, of course. Because, as we said, the first thing that gets affected is zone 3 because it has low blood supply and low nutrients, low everything. What's the possible solution? Is giving this in acetylcysteine to increase glutathione or reduce glutathione. Because if you have lots of this, it will neutralize the free radicals even more and more, ending up with no damage. Okay. This is a summary for a few diseases and whether it was conjugated or not. As far as I know, the doctor said no, you don't need to remember each problem with each syndrome of these, but just know that the general idea. So when it's conjugated, as we said, it will be obstruction or one of these syndrome, Rotor or Durbin-Jones. Okay? I will talk more about the diseases in a minute. Unconjugated, as we said, it's either hemolytic anemia or could be also some diseases such as Krigler-Najjar and Gilbert, and we'll be talking about them also. We have our mixed jaundice, which is basically any problem that happens with the liver. Why do we call it mixed? Because both are impaired. The liver is our main thing, okay? You can't afford to lose it. If it's lost, you don't talk about unconjugated, conjugated. Both will be elevated, both will be gone. There will be a lot of problems that are hard to interpret, so we don't talk much about. Now for physiological jaundice, it's what we call, first of all, this is unconjugated bilirubin, okay? And it's very common, and we call it physiological, not pathological, because it's normal. 50% of newborns have this jaundice. What do we mean by that? It's not that the baby is born yellow. If it's born yellow, this is hyperbilirubinemia, and it's a problem. But if it's for 2 or 3 days after the baby is born, he starts becoming yellow, and then it goes away, okay? Then that's okay. It resolves on its own, and this is what we call physiological jaundice. What's the reason? Please note it. Immature, I will not write it, but just remember. Immature hepatocytes. Their liver cells are not functional yet. So they can't conjugate, they can't execute, they can't do anything. So this is what causes physiological jaundice. Please remember it. This is a summary just to know for the diseases, to know that any impairment in any part of the pathway will cause a problem, okay? These are the serum markers. The doctor said that you just need to know this. Alkaline phosphatase is higher in what? Biliary obstruction. this is the clinical correlations that we will talking about. And as we said, cholestasis will be, we will have our high alkaline phosphatase. You don't need to remember the details, but just know that, for example, here in Krigler-Najjar, you have, do you remember UDP glucurylase transferase, the one that we use to conjugate bilirubin? It will be impaired here, or we have deficiency. Either type 1 where you don't have the enzyme, or 2 where it's reduced. You don't need to remember the details. Like the doctor will not give you a clinical scenario and tell me, is this Krigler-Najjar, is it Gilbert syndrome? But if I were to recommend you something, just know which one is conjugated and which one is unconjugated. Try to link it on your own, because we don't really know much about diseases yet. Now this is summary for jaundice types, as you can see. If it's pre-hepatic, as we said, it will be hemolytic. We'll have high red blood cell breakdown. What about unconjugated bilirubin? Elevated, conjugated is normal. Is there bilirubin in urine in pre-hepatic? No, because the problem is pre-hepatic. Conjugate bilirubin is normal. So the bilirubin that ends in the urine is like non-existent. It's normal. Now for urine urobilinogen, as we said, it will be more. This is just a summary to know. And I want you just to know the stool color because we didn't talk about it. As you can see, it will be normal or dark in unconjugated bilirubin, because you will have your stercobilin happening normally. But for post-hepatic, okay, here, it will be clay colored. Why? Because you don't have the stercobilin, which gives the stool its color. How is that? Do you remember here? Where did we get the stercobilin from? Did I miss the slide? Yeah. It was here, right? And in conjugate bilirubin, you have this problem. The blockage is here. So you are not reaching the stercobilin, which will cause pale or clay color stool. Yeah. Okay, so for the 12th lecture, inshallah, it will be an easy and quick lecture. There is nothing much to know here, but yeah, it's large intestines motility. So, to start, I just want to set up something that what we call segmentation in everywhere else in the intestines, like the small intestines and all of that, is here called hostra or hostration, which is basically these small buckets. Can you see this is a hostra, this is a hostra, this is a hostra. And their job is basically to separate and make segmented feces. So it's not a continuous part. No, it's basically segmented. Does that make sense? And they do that by organized contractions and all of that. And they can be propulsion, which is the difference between segmentation and hostra is that they can be propulsion or retropulsion. Propulsion is meaning it's moving forward like this, and retropulsion means it's moving backwards. So that's not something that you see in segmentation, in the norm segmentation. Now, the basic electrical rhythm. What's the basic electrical rhythm? It's basically the slow wave frequency. Do you remember the waves that we talked about? It sets basically the maximum action potentials that this part of the intestines can have. And the highest BER that we have in the intestines is in the rectum, which is 70 minutes. Why? Because the rectum should always be empty, and we will discuss that later. Either the feces go out or they go back into the sigmoid column. We will talk about that. Now, the peristalsis and mass movement. Do you remember when we said segmentation is hostration here? Peristalsis is called mass movement in the large intestines. And there is a question about this in the 17th fine. In the large intestines, it's called mass movement. Why is it called mass movement? Because peristalsis is tiny movements. In the large intestines, okay, the movement is happening by 20 centimeters per moment. Can you imagine that? So we are moving 20 centimeters of material, each one of these mass movements. And it's happening 3 to 4 times a day. So only 3 to 4 times a day, and it's enough to empty this large intestines. Can you imagine? SubhanAllah. Now we have to talk about this, fibers and transit time. The fibers, basically what's fibers, just to set it up. Fibers are material that you eat that cannot be digested, like cellulose of the vegetables, and it's mostly things in the vegetables. So the more fibers you eat, the less transit time you have. What's transit time? it's the amount that food takes until it's excreted out. Does that make sense? So when you eat more fibers and you eat more generally, you have lower transit time, means the large intestines will try to excrete faster. This is why when someone eats a lot of fibers, they will have a better health of their large intestine, because the food is moving faster. Why do we want the food to move fast? Let it sit. What's the problem? I'll tell you what's the problem. We have bacteria in the large intestines. It's not a sterile part of your body. And the problem is these bacteria usually live on they ferment carbohydrates and proteins. Now the fermentation of carbohydrates, it's okay. We can afford it. There is no problem with that. Okay? The problem is that protein fermentation gives us these guys. Very toxic stuff. Okay? So we don't want the bacteria to ferment the proteins. Okay, so what now? The carbohydrates, the bacteria usually loves the carbohydrates more. So they will start by fermenting what? The carbohydrates. But when the feces stay a lot of time in the large intestines, the bacteria will finish the carbohydrates and they will start fermenting what? Proteins. This is why you want the feces to move as fast as possible before the bacteria can finish fermenting carbohydrates. Okay. Now for the water balance, I just want you to know something. We have 9,000 milliliters of fluid entering the intestines. The small intestines, I mean. The small intestine absorbs 7,000. What's left for the colon? 2,000. The colon absorbs 1,900 and leaves 100 to be excreted out of the body. Now, I want you to remember that the colon can absorb more. So what if it's not 2,000 that's reaching the colon? What if it's 4,000? Okay, it has the capacity of 5 liters of absorption. So yeah, there is no problem even if you increase. But that's only if you increase like a reasonable amount. But when it's, for example, 6,500 entering the colon, it will reabsorb until its capacity is fulfilled. But the rest will be excreted. So you will be excreting this amount of water. Why is that a problem? Diarrhea, basically. You will be having diarrhea. So I want you to keep in mind that this is the good amount of water that should be kept in the feces. If it's more than that, it's diarrhea. If it's less than that, it's constipation. Also, if the colon starts secretion, and this is what we call secretory diarrhea that we have talked about, even if the amount that's reaching is normal, but the colon is not even absorbing, it's secreting now. So what? It will be, also it will cause diarrhea. And I want you to keep in mind that the main function of the large intestines is to absorb water, to make the feces suitable for excretion. Now, sodium transporters. Here I want you just to know that we have 3 transporters that are mainly causing the sodium absorption in the large intestines. The main one being the sodium channels, the normal sodium channels. Or it could come with other molecules such as sugar and amino acids like we discussed before. Or it could be by this antiporter. This one is the most important, okay? This is a summary for the transporters. I don't think he will be asking about this in any ways. Now, this is a very important concept that had a question about it before. And basically, have you ever eaten and once you eat, you feel like, okay, I might need to go to the bathroom? But that doesn't make sense because when you eat, the food is now in the stomach, right? Why would you feel like you want to go to the bathroom? It's not in the large intestines. This is what we call gastrocolic reflex. Gastrocolic reflex is a connection because of neurons and like your brain basically connects your stomach with the large intestines. If there is food in the stomach, the distension of the stomach causes this gastrocolic reflex, which causes mass movement in the large intestines. And mass movement is like 20 centimeters or more, and it causes you would want to go to the bathroom. Now, we have here what we call ileocecal valve. I want you to know that the large intestines are a non-sterile environment. They are filled with bacteria. The small intestines is, let's say, it's mostly sterile, okay? So you don't want bacteria to go back. So what do? We have this valve between the ileum and the cecum, what prevents the retrobulging of food or the feces, into the small intestines. What if the valve is incompetent? You will have this condition, which is self-explanatory: small intestinal bacterial overgrowth. Now, for the rectal motility, you will see that once the food reaches the rectum should always be empty. The rectum will try one of two things. It will try to go out. First thing, we have two sphincters, like in renal, if you remember, the internal and external. So we also here have internal anal sphincter and external anal sphincter. The internal is involuntary, so it will open. The external is voluntary, so it's up to you. If you tell the external to contract, it will tell the rectum, no, not now. So what will happen? The food will go back. Retrobulging movement to the sigmoid colon. But, sorry, not the food, I'm still saying food. Feces will go back. So this part of the rectum will always be empty, either forward or back. So, but what if, okay, it's appropriate time to defecation. So you tell the rectum, okay, now we can, it's appropriate time. The external anal sphincter will relax and let the feces out. Now, the external sphincter, I want you just to know that it's controlled also by the pudendal nerve, the same one that controls the external urethral sphincter, if you remember. And yeah, it's voluntary. Once I want you to note the pressure, what's happening here in the rectum. The feces are coming and they are increasing the pressure in the rectum because the rectum is trying to defecate. Same thing with the internal anal sphincter. You will see it's starting to relax to allow the it's like the detrusor, internal urethral sphincter and external urethral sphincter, if you remember that. Yeah. So it relaxes. Now for the external anal sphincter, it will automatically, like, subhanAllah, you're born with this reflex that it will contract and wait for your order. Okay? The voluntary control. Now the voluntary control either delay. If it's delay, it will cause contraction, and okay, we'll maintain the contraction until the time is appropriate. Or it's okay, we can defecate, so it will relax, and so will the puborectalis muscle. I want you also to keep this muscle in mind. Why? Because the puborectalis muscle is this muscle that's hugging the rectum. Can you see? It's squeezing it. So if you need to defecate, it should relax. So it also acts the same way the external anal sphincter act. So yeah, and this is why sitting and squatting is the most optimal for defecation, because, like, physiologically speaking, because you are relaxing these muscles and allowing the rectum to empty. Now, if you have poor motility, the feces will stay much more in the rectum, and this will cause hard feces and constipation. If you have excess motility, there will be diarrhea because less absorption. Hirschsprung disease, you know Hirschsprung. It's not new to you when you have a ganglionic segment. And yeah, thank you so much. I hope this was helpful. Please don't hesitate to reach out to me if there's any problems, any mistakes, anything you didn't understand. Here's my number, here's my email. I would suggest to you guys, please, after understanding everything here, go solve Dr. Angelica's box questions. They are very important, and she usually gets similar things. And yeah, I hope you all good stuff, guys, and see you inshallah.